A very greeting to one and all present here. Welcome to this webinar. It is my immense pleasure to be a part of this 1 week live webinar series of International E-Conference in Plant Sciences organized by the Department of Studies of Botany, Davanegere University in association with one of the university in Mexico. I am Dr. Sinosh Skariyachan, Assistant Professor, Department of Microbiology, Pius X College Rajapuram, Kasaragod, Kerala. And I would be delivering a talk on deciphering the role of or the relevance of computer-aided drug discovery, prediction of three-dimensional structures and folding pattern of molecular target. And this topic has got lot of relevance as we are living in the era of COVID-19, and in this situation you know that today 26th May 2020, as per the statistics of WHO is concerned, there were 5.4 lakh confirmed COVID-19 cases with 3.46 lakh of death. And earlier the WHO declared this one as public health emergency of international concern, and later by seeing the severity of this particular condition, the WHO has declared this COVID-19 as pandemic in 2020. And as per today's statistics are concerned, our India, now we are in 10th position with 1.45 lakh confirmed cases and 4,167 deaths. And you know, there are lot of companies in an international level. They are trying to develop vaccine, even some antibodies and some lead molecule to combat COVID-19. And if you look this particular scenario, you often find one of the most important area playing in this particular drug discovery of COVID-19, that is computational biology and bioinformatics. And you can see I have shown two paper. One is the Nature paper, another one is the Science, and both the papers are very recently published. And this paper says the structure of the major protease P-Pro from COVID-19 virus, discovery of its inhibitors. The second one is the crystal structure of SARS-CoV-2 main protease, provides the basis for the designing improved alpha ketamide inhibitors. So what I am trying to tell is the importance of vaccine development. Currently it's a most popular need, and in this slide you can see that I have taken last 2 to 3 week research work. Past 2 to 3 week research work, when I went to the literature, I could see most of the internationally reputed journals, they are publishing lot of papers about COVID-19 research. And what these papers are about? All these papers you can see one peculiar factors, that is drug discovery by using computational biology. Here one journal, Travel Medicine and Infectious Disease, this published by Elsevier with an impact factor of more than 4. Similarly, International Journal of Antimicrobial Agents, and another one is the Emerging Microbes and Infection. And here you can see the cell discovery and viruses and chemical physics letters, and all these journals. Not only this one, there are many other journals. They publish papers based on COVID-19 research exclusively on computational biology, and that is the power of computational biology and bioinformatics at this point of time. Rather than looking for any aspect, the scientific community obviously knows that computer aided drug discovery, identification of potential molecular drug targets are one of the most important aspects to combat COVID-19. I mean, to find out novel drugs against COVID-19. So, what we are going to see here is all the scientific community, whatever I have shown in the previous slide, they have developed, they have worked on certain structures. You know that the novel coronavirus, SARS coronavirus 2, that is an RNA virus that was from 5 to UTR to 3 UTR. There are a lot of genes. It's an RNA virus, and the RNA, from the RNA the cDNA will be synthesized, and the cDNA will produce lot of proteins, the major surface proteins and non-enveloped proteins and enveloped proteins, and similarly non-structural proteins, and some of them are having the crystal structures and some of them are not having the crystal structures. But scientific community using bioinformatics and computational biology, they identified these particular target, that is molecular target, can be act as the potential molecular targets for the vaccine development. So in the absence of an experimental structure, of course, that is crystal structure, the scientific community tried to create lot of computational models, and that models they used to validate by using bioinformatics as software. So that is the importance of computer aided drug discovery. So why I have started with the COVID-19? COVID-19 is the one of the potential concern. Here we are using the fundamental aspects of computational biology and drug discovery for finding a novel drug target. And now we will go to the computer aided drug discovery. Computer aided drug discovery, it is a very complex protocol, and this will take a lot of time, and a company should spend a lot of money and lot of infrastructure, lot of effort this company should put. Why we are not able to develop vaccines? COVID-19 we are having a lot of there are a lot of companies are trying to develop vaccines against COVID-19. Vaccine development is not so easy, and now people are approaching the repurposing of the drug. There are, you know, that hydroxychloroquine, even our Indian Council of Medical Research is given green signal for the usage. Chloroquine and lopinavir, ritonavir and remdesivir, all these drugs are already discovered, and they have developed for various viral as well as parasitic infection. Now they are trying to repurpose the drug. Why they are repurposing? Now current drug discovery protocols are very tough to go ahead, and for conventionally speaking, if you wanted to develop a molecule, if you start compound is 1000 compound, and there is a initial pipeline, then it may take then after preclinical level it may reach 250 compounds, and that will take a minimum 6.5 years, and the clinical level, in the clinical trial only 5 compounds goes, and finally the FDA, the company is very lucky, the FDA may approve one drug or so. And here the time it is required for nearly 15 to 16, 17 years with a lot of initial investment. That is the problem with the conventional drug discovery sector. Now, as I said, here you can see the drug discovery scenario. This is a drug research area. Initial there are 10,000 compounds for testing, the preclinical, the lab and animal experiment. Only from lot of molecules will be screened, and finally those molecules are qualifying certain type of the druggish properties or pharmacokinetic features and toxicity features, finally only a few molecules will be coming into the later stage. Then if you go to the phase 1, phase 2 and phase 3 clinical trials, then the phase 1 it is 20 to 100 healthy volunteers, and phase 2 it's that they are scaling up actually 100 to 500 patients, and phase 3 1000 to 10,000 patients with the efficacy and adverse side effect they will check, and there will be very limited number of molecule will be qualifying all these features finally, and it will take a lot of time, nearly 2 years finally. When it is reaching to the phase 4 clinical level, it will take a lot of years. Totally a company you need to spend more than 1 billion euro to in order to get one drug into the market. And now the problem here is, if you know the disease means any disease, you know that there will be a key player of the disease, and that disease will be having the expression that is happening due to the expression of mainly from so many molecular target, mainly they are protein. So you need to isolate the protein and purify the protein, and identifying the interaction between the protein in that disease, and all those things will take a lot of years. So that is why it is a time consuming process. Now identify the protein from the disease. Now this is the protein. Now you are having some set of drugs you will be identifying from small molecules, and that it will take 2 to 5 years time, and that will go to a preclinical level. 1 to 3 years it will take, and later it will go to the clinical trials, and it will go to, as I said in the previous slide, so it will go to lot of testing finally in the lower animal, and mice, rat, in the later higher level of organism. Finally it will reach the formulation and scale up, and finally they will try to file a patent, and finally in the human clinical trials it will take a lot of years, and the company is so lucky they will be able to come up the drug. So all that slides whatever I am trying to tell is it is a very very complex process. It is very complex process, the discovery of the drug. That is the complexity associated with the discovery of the drug. Then if you look this particular slide, and you can see that, so if you go to the previous slides, so the initial phase, so all the cases, the initial phases are the one in which we are dealing with so many targets and so many lead molecules, and it is very difficult to set up an experimental level and very difficult to study in experimental way. And conventional drug discovery, as I said, it has a target identification, target validation, lead identification, optimization of the lead, and preclinical and clinical studies. And the initial level, that is the target identification, target validation and lead optimization, lead identification and optimization is the major aspect. A lot of time and lot of monies people are spending. So why can't we use bioinformatics and computational biology with accuracy? We will be able to screen good molecule, that is a drug with ideal drug like characteristics, pharmacokinetic features and toxicity. Why lot of molecules are failing? It is very difficult to identify a drug molecule with all these peculiar parameters. That is why the molecules are failing in the later in the clinical level. And why can't we use the computer aided drug discovery? The consisting of 3 approaches. The structure based approach, there so many modeling aspects, the prediction of the structures, the docking studies and molecular dynamic simulation studies, QSAR modeling, computational chemistry and chemoinformatics, molecular profiling, network pharmacology, network analysis, so complex. So that is why I told we can use computational biology for this particular study, and you will be able to implement computational biology for the rapid screening of molecular target. And whenever you talk about a molecular target, most of the molecular targets are protein. So for the student community, for the sake of students, now I will be dealing with some basics about the molecular targets, mainly the proteins. As you all know, proteins are the building blocks of the body. They are the building blocks of the body. They are the linear sequence of amino acid which are connected by one amino acid, are connected by a peptide bond to the next amino acid. So you know that amino acids are alpha amino substituted carboxylic acid, and they have an amino terminal end and they have a carboxy terminal end, and most of the molecules are different in their side chain. And the formation of this long chain amino acid, it requires a strong rigid planar covalent bond called a peptide bond, and this peptide bond is the major bond that stabilizes the amino acid structure. And there are 20 amino acids. You know that there are lot of classification systems are there for the amino acid. These 20 amino acids are there. They can be aliphatic amino acid, hydrophobic amino acid, aromatic amino acid, positively charged amino acid, polar amino acid, charged amino acid, and small amino acid. And what is the main thing? As I said, they have a carboxy terminal end at one end and an amino terminal end at the other end. And the major difference is the side chain. Some amino acids have a normal linear side chain, and some amino acids are having, say for example, amino acids like tyrosine, they have an aromatic heterocyclic ring as the side chain. And the side chain is the main important difference in the case of amino acid. So in order to study this protein chemistry, the molecular target, one should understand the biochemistry of this protein thoroughly. So you know that proteins are isolating from the beautiful concept protein synthesis, the central dogma of biological sciences. The DNA is transfer is message into mRNA, and the mRNA produces the transcriptome, and that moves to ribosomes, and there the synthesis of protein will take place. And the protein, the amino acids are the basic building block. That amino acid we can represent as a codon, individual codon from the mRNA, and these amino acids we can represent by single letter code. And the single letter codes are very important. One person is dealing with the bioinformatics and computational biology, because bioinformatics, the amino acid sequence we are representing in the form of a single letter code. Say for example, phenylalanine, immediately we should know that is F, lysine that is K, so like that, and Y tyrosine, and W tryptophan. All the single letter codes are coding for the amino acids. So that is why the single letter codes are having very important role in the protein chemistry. Now I told you the peptide bond, and the proteins are having the unique sequence of amino acid, and they are stabilized by the peptide bond. And this peptide bond consisting of three types of bond. One bond is out of three bond, there are two are flexible, they are rotatable. One is non-rotatable. That non-rotatable bond is known as omega, that is C1 and N. The bond between C1 and N is known as that is omega, and the bond between C1 and the C alpha, that is a psi, that is rotatable, and this is the C alpha and N, that is a phi angle. And these torsion angles are very important. So one particular peptide bond will be having two torsion angle. So you will know that both the torsion angles are flexible. Then you can imagine how many flexibility one particular protein can produce. How much the protein will be flexible. So if an amino acid is of 100, then if a protein of 100 amino acid in length, then if you check all the possible conformation that particular protein is going to give, that is a very huge conformation. Every peptide bond, there are two dihedral angles. Then the total number of conformation, the flexibility, if you count, that will be 2 raised to 100. That is a very huge number. So because of the dihedral angle, the proteins has got a huge amount of degrees of freedom. So then the protein has a linear, uh, they have a linear sequence. That sequence are coming from the sequence, I told you, a DNA sequence, and that particular DNA has a gene. That gene will be having, after transcription and translation, it will be produces to the protein, the amino acids, and the gene code for a specific amino acid, and these specific amino acids folded into a very unique and specific way, that is known as folding, and that will fold into a particular protein, and each protein has a characteristic 3D structures, and that 3D structures is clearly giving you the function, and that function is the ultimate reason for the mechanism and the metabolism of all the organism, including human being. So there is a proper relation between the gene, proteins, and its function. So starting from a sequence level, from the basic level to the advanced level, that is the power of bioinformatics. Here the bioinformatics, we will talk the molecular biology in terms of the basic amino acids and the nucleotide level. So you know that in the case of an organism, proteins are the building blocks, and the basic building blocks are the amino acids, and you know that in the case of a DNA, the basic building blocks are the ATGC code, adenine, thymine, guanine, and cytosine, and that code for triplet, and that will give you the amino acid. So that is the speciality of bioinformatics. So all other science that generally start from a cell, but bioinformatics start from the ultimate molecular component, that is the molecular fossil of the organism, where the DNA and the proteins. So that is the power of bioinformatics. Then how the protein function? The protein function due to the spontaneous folding, and the folding assume a particular three-dimensional features, three-dimensional folds, and each fold they produce as a typical function. From a linear chain, you can see a fully folded functional protein you can see. Then the protein folding, it was a very long history of studies actually, and so many people have studied, and one notable study is conducted by a great scientist called Cyrus Leventhal, and that concept is known as Leventhal paradox, and this is very interesting. The protein can fold so many conformation, and if you give nanosecond time, within nanosecond time the protein can spontaneously fold, and every nanosecond, this nanosecond the conformation it is different, and this nanosecond the conformation is different, and the third one the conformation is different, and like that so many nanosecond. If the protein start folding, you will be getting the folding time, you will be getting lot of conformation, and you know the time required to take the entire folding of the particular protein is the bigger than the age of this particular universe. That is what is known as Leventhal paradox. And this particular folding pattern, and what is happening in the folding pattern, I told you there are so many conformations the protein will produce, but not all conformations are responsible for the biological activity. Only one or two conformation is responsible for biological activity. That is the most complex problems in the biology. When which is that particular conformation that produces the actual biological function, and out of so many conformation, which conformation produces the biological function? It is a very tedious process. So protein folding kinetics. So this is a typical protein folding diagram. Initially when a protein start folding, what will happen is they will acquire the secondary structures, and the secondary structure try to fall, and their energy, the thermodynamic energy will slowly released, slowly coming down, and finally what will happen, they slowly coming down, again slowly coming down, and finally that particular protein will get a least amount of energy, and there is a sudden hydrophobic collapse from the unfolded form, and in between there is an equilibrium state that is known as molten globule, and from the molten globule it reaches to the native state where it reaches the maximum lower energy, where it fully stable, functionally stable, and that is the kinetics of protein. Kinetics of protein, that is very interesting. So when you apply the bioinformatics concepts also, the protein folding kinetics are very important. When a protein start folding, initially it will start folding, and which stage the protein will be able to produce the least amount of energy, that particular conformation we call it as bioactive conformation or minimum energy conformation. And in our body, that will be the conformation they perform the actual function. And if the protein misfolding, you know that the one particular protein, one amino acid is misfold, it will create a lot of problems. And you know that the free energy changes, that will create a lot of impact in the biological system. And a simple example, sickle cell anemia, you know, in the position 6 there is a replacement of valine and the glutamic acid, there is a simple change, and that leads to the sickle cell anemia. Similarly, you can see that so many protein misfolding diseases are there. So that is what is the importance of the concept protein folding. Now when you talk about this protein folding, then you can see the proteins are start folding spontaneously. Then initially they will start folding, then slowly they will acquire its conformation, and they will try to acquire, they will come to their energy level, then very less energy level they will try to come, and they will initially then unfolded form of the protein that will produces the unfolded form of the protein, and slowly they are slowly start folding. And you know that you can see in this small video there is a polypeptide that is start folding. Actually they see 1 nanosecond time, if you allow to simulate the protein, it will try to fold. And you will see the steric clashes, they are trying to adjust the steric clashes themselves. The secondary structures are adjusting themselves, they are fitting themselves to the actual native structure. They're slowly they are adjusting the energy. Finally it fully folded, the most energy minimized form it has come, that is known as bioactive conformation. Now the structure of proteins are very important. I told you, if you wanted to study the function of the particular protein, you require the three-dimensional structure. So there are a lot of conventional approaches are there, the three-dimensional structural discoveries, X-ray xerography, NMR spectroscopy, similarly electron microscopy. Very recently 2017 Nobel Prize thanks goes to Dr. Joachim Frank and his team. He got Nobel Prize in the discovery of the latest microscope, that is known as cryo-electron microscopy. So it is very essential to find out and very difficult to find out a very good angstrom, closer to 0 angstrom root mean square deviation accuracy structures. And this identification of the structures, it is very tough task, because most of the protein, the crystal structures are not available, but their sequence are available, and the crystallography NMR facility, it will take a lot of time to do it. And you know that cryo-electron microscopy is the latest approach. Somebody require a lot of initial investment, and even if you are having all those things, except cryo-electron microscopy, by using other approaches we cannot crystallize or cannot find out the structure of all the three-dimensional structure of the protein. Say for example membrane protein, they are highly hydrophobic, you know that. So it is very difficult to crystallize membrane protein. Then what is the importance? So if you have a very good structure, then only we are able to study the function. That is the important thing. Then coming to the structural aspect, all of you know this is very basic thing, since this will be useful for this so many students and research scholars. So I will be dealing with some basic concept about the protein structure. Protein has got 4 different types of structural organization. The primary structure, secondary structure, tertiary structure and quaternary structure. So the importance of this all structural level studies you can look by simply looking one aspect. All these structural studies have got the most prestigious award in the world, that is Nobel Prize. So primary structure, so here you can see the linear chain of the amino acid sequence. They are connected by peptide linkage. The amino terminal end of the amino acid will be interacted with the carboxy terminal end of the amino acid. There is a formation of beautiful water molecule will takes place, and you can see that a dipeptide unit, and there is a formation of a peptide bond. And this study has conducted in the bovine insulin, the structural elucidation of bovine insulin by a great scientist called Dr. Frederick Sanger. When he was just 30s, he got a Nobel Prize for the discovery of the elucidation of primary structural aspect of bovine insulin. Then if you see this primary structure, if it is twisted in a particular axis, additional forces coming into picture. The side chain, I told about the amino acid, there is a side chain. So the side chain groups are interacting, and they forms interacting each other, and they fold spontaneously by two main way. One is alpha helical structure and another one is beta platelet structure. In alpha helical structure, the polypeptide chain is folded in the form of a typical alpha helical structure, and in the case of their right-handed alpha helical structure and left-handed alpha helical structures are there. There are so many other types of structure. We will see that all those things later. And the beta platelet structure, you can see this form sheet. There are multiple parallel amino acid chains amino acid sequence will be available. The one amino acid will be interacted with the nearby the other amino acid by hydrogen bond. So what is the most important force that is stabilized by this structure? That is hydrogen bond. So in the case of secondary structure, the hydrogen bonds are the most important structure that is stabilized. So they have two types of forces. One is the hydrogen bond, next one is the peptide bond as usual. Now the next important aspect, this again as I said, gone to the secondary structural discovery by the great study conducted by the scientists, the structural elucidation of collagen. They have identified the great scientist Dr. Carl Linus Pauling and Robert Corey and Herman Bradson. They got the prestigious Nobel Prize for the discovery of the secondary elucidation of secondary structure and structural aspects in collagen and other structures. And the next one is the tertiary structure of the protein. Tertiary structure of the protein is the fully folded one functional unit of the protein. Secondary structure, this again if it is start folding, what will happen? It will produce a tertiary structure. You are having, and these tertiary structures are producing by using weak forces such as hydrophobic interaction, van der Waals interaction, hydrogen bonds and ionic bond, on all other types of interaction. So it become a fully folded form. Here you can see the structure of myoglobin. They are having myoglobins are a muscle protein. Here you can see a ligand at the center. There is a ligand at the center, and you can see myoglobin is fully alpha helical structure. There is no almost everything is fully converted into alpha helical structure. So and fully folded myoglobin structure and the tertiary structure, and this is a fully functional subunit called domains. So a concept called domains, a fully functional subunit of the protein, that is known as domains. So that is the great study conducted by Dr. John Kendrew. Again he was awarded the prestigious Nobel Prize. Then the next structure, as usual, you know that hemoglobin, the structural the concept of multiple dimers. The concept of dimer, here it is a heterotetramer. It consisting of 2 alpha subunit and 2 beta subunit. You can see 2 alpha subunit. This is subunit 1, subunit 2, subunit 3 and subunit 4, and they are having a central porphyrin ring, and they are connected to the histidine amino acid of the particular chain, and we call it as this each chain, the domains and each subunit called as one chain. This is one chain, this is second chain, third chain and fourth chain. This is a beautiful study conducted by Dr. Max Perux, and he also got the prestigious Nobel Prize for this particular studies. Then the relevance of structural prediction. So why do you want to predict the structure? I told you, in order to study the functional aspect of the protein, you require the structural studies. And it was a nearly a 4-decade scientific community has studied, and it was a very big task in biological science. Okay, and it was a primary motivation of computational biologists. And still it is an open problem, even though we are having X-ray crystallography, NMR spectroscopy, the latest cryo-electron microscopy. It is a very tough to identify the structure. It has so many complexities, so many issues. Crystallizing the protein is very difficult. X-ray crystallography will not work in membrane proteins, and the NMR is the best method for that. And all those things, it requires a lot of effort, and that is why here comes the role of computational biology. And not only that, the current there are a lot of limitations. Then the you know that the experimentally solved structures, where you will be getting, there is a database developed by Research Collaboratory for Structural Bioinformatics called PDB, and that PDB has a protein data bank, and they are having very less number of nearly 1.6 lakh structures are available in PDB, and a lot of structures, 95% of the protein, non-protein, the 3D structures are still it is not known. There is a huge gap between the sequence and the structures, and in order to study the engineering and their function of a protein, the structural aspects are very important. So that is why this particular structural studies are very important, and that is why it is very powerful approach. The computational biology, very powerful approach. So what is the role of a computational biologist? So what is the objective and what is the aim of the computational prediction of the molecular target? Should get an accurate 3D structure that should have even if you are predicting, it should comparable with experimental structure. If later somebody discovered, somebody crystallizes the structure, or somebody identify a very good cryo-electron microscopic structure, our predictor structure should have a very good accuracy to this particular structure. It should have, and you should know, as I told, the folding problem. You should know the folding problem, should know, understand the folding problem, and you should go to the black box of the plane, and that is the black box of the protein chemistry. That is the folding problem. And I told you, even 25% of the protein will not be able to crystallize, and it is too big for NMR, and thus because of all those things, you need to develop a lot of protein engineering exercises, lot of drugs, lot of molecular target, enzyme substrate relationship you need to study, lot of new things you need to study by looking the 3D structure of the protein. So still the protein prediction is a very big hurdle because it requires lot of energetics. So it is very important. The energetics are lot of energetic. It is computationally, it is very expensive. So, and conformational search possibility, I told you in the beginning, it's a Levi-Dahl paradox. If a protein, one peptide unit has two dihedral angle, torsion angle, psi and phi, and a protein has at least 100 molecule, 100 amino acid residue in its length, then you imagine the total number of conformation in that particular protein is going to produce, that is 2 raised to 100. It's a very huge number, and that is what I told. It is a very huge number, that is longer than the time of universe to search. So that is what Levi-Dahl paradox says. So it is very difficult to identify. Not only that, and all those conformations are not always gives the functional relevance. They are not bioactive conformation. You require bioactive conformation. So these are the next important thing. Then, so firstly, one should identify the structure from its primary level. Understanding the primary level, the basic amino acid sequence. So you can see that the primary structure analysis is the fundamental approach for a computational biology person. Before they are going to model a 3D structure, the fundamental, the basic information about the protein, it is very much essential. The amino acid composition, the atomic composition, the molecular weight, isoelectric point, peptide mass, obviously the post-translational modification. And we are having two important methods for primary structure analysis by computational biology. One is the protein identity. Based on the composition, we can identify identical proteins, And next one, based on the physical properties, we can identify the similar type of sequence of the protein. And how will you do this? We are having a beautiful bioinformatics tool called XPaSI server. This XPaSI server is known as Expert Protein Analysis System. This is the a screenshot. What you are seeing in the screen is the XPaSI server, and just recently I have taken, you can see a XPaSI server. You will be able to get a lot of resources. Whatever you want to do in proteomics study, you can do XPaSI. It's a beautiful server. Starting from the very preliminary level, you will be able to go into a highly advanced level. Even you can data such as the mass spectroscopic data, you can analyze protein folding, you can predict it. You can study phylogenetic analysis. You can study the disordered regions of protein, predicting the primary, secondary, tertiary, quaternary structure of the protein, and you will be able to predict the post-translational modification and so many. And now you can see they have launched a special division for COVID-19. So you can see all the bioinformatics data. That is the aspect. Bioinformatics is today's science. What is happening to the world currently? Today, what has happened? You will be able to see such a kind of information by referring the bioinformatics databases and the tools. So by looking this one, you can see, and I told you there are two important approach: protein identity based on composition and the physical properties based on the sequence. There are a lot of bioinformatics tools. This simply you will be able to see by looking this server. It is very, very simple. Only thing is, like, you should know the basic amino acid sequence what you are going to work. So that is the only thing. So you are having the tool for under this category: a comp ident, this amino acid composition and identity; a comsim, amino acid composition and similarity; prop search, that is property search; then tag ident, okay, mouse molecular weight search. Similarly in this category, protparam, protein parameters; compute pi, you know that isoelectric point is one of the most important things as far as protein is concerned; peptide cutter, T-grease, and statistical analysis of protein sequence. All those tools you can use for primary structure analysis. So I will tell you one simple example, the protparam tool. So protparam, this is the home page of protparam. If you open the XPaSI server, you will be able to see protparam, and here you can see there is a box, or you can paste the sequence into the into the particular form. And what format you will paste, and there are databases like GenBank, DDBJ, EMBL, UniProt. So all these databases you can retrieve the sequence in the form of a particular format called FASTA. So bioinformatics tools and softwares, they require a specific format to recognize the sequence. That is FASTA format. What is FASTA format? The sequence that is starting with a greater than symbol and some ID numbers will be there. That will be from GenBank or UniProt or ENA or DDBJ or any other ID you can see. And here you can paste that particular sequence, and what you can do is and you can just you will be able to get the result. So here you can see the result. So this is the result of protparam. Here what you can see, just click and it will take 1 second. That's all. Here you can see the amino acid. I have submitted a query sequence of streptolysin. That is a protein. Since I am basically a microbiology person, I work on bacterial proteins and antimicrobial resistant genes and all those things. So streptolysin gene I have submitted, and you can see it has a thiol activated cytolysine, and it has 579 amino acids. So whatever preliminary information you want, the number of amino acid, molecular weight, and theoretical isoelectric point, and concentration of each amino acid, concentration of what is the percentage of alanine, arginine, leucine, valine, tryptophan, tryptophan, what's the number of carbon, hydrogen, nitrogen, oxygen, any number, what is the molecular formula, what is the extinction coefficient, and lot of, and what is their half-life. All those information you will be able to get from primary structure. Another tool called peptide master, the other category. So here also the same steps. So you can download or retrieve a protein sequence from the database. If you don't have a protein sequence, then what will you do? You can have your own experimental data. You might have crystallized, or you might have analyzed some sequences. Okay, some sequence you might have analyzed. You might have sequenced some particular protein, or you are having a small gene, and that you can convert it into a protein. That also it is possible in this bioinformatics tool. So that you can do. So here what peptide mass tool will do, here you will submit the ID, the same protein streptolysine, and you are having a drop down list here, all the possible enzymes. Here you can see on that particular enzyme which enzyme you wanted to select. So you wanted to select trypsin. There are similarly you are having chymotrypsin and so many enzymes, papain, okay, or pepsin, or so many enzymes you can see. So which one you want, you can click, and this particular sequence will digest the query sequence. This particular enzyme will digest the query sequence whatever you have submitted actually. So that is the that is what it do. So here you can see the output. This is known as peptide mass output. Here you can see this is the it is shown in the red color, whatever it is shown. So here you can see this is the specific site that is for these enzymes. So and you can see there is the bigger fragment. It will come first, then comes the next one, then comes the next one. Here the region also they will tell from which region to which region these peptides are present, which region to which region these particular peptides are present, what is their size and all those things, the preliminary information they will tell. So this is about peptide mass. So this is the way you can, lot of primary structural analysis tools are present. Then comes the secondary structure. This is the most important structure of a particular protein. If you wanted to study any protein in the functional aspect, you should study the properly about the secondary structure. So as far as the secondary structure is concerned, they are the backbone of the protein. So these secondary structures are the two types: regular secondary structures and irregular secondary structures. So regular secondary structure, you know, alpha helical structure and beta pleated structure. Alpha helical structure, as I said, you are having several types of alpha helical structure. Protein chemistry you know very well. That is the molecular biology. You can see the biochemistry, you know, the alpha helical, right-handed alpha helical structure, left-handed alpha helical structure, 3-turn helices, pi helices, 2.2627 ribbon. Okay, so many types, and beta sheets, parallel beta sheet, anti-parallel beta sheet. So all those things are regular secondary structures. Then you are having irregular secondary structures also. These irregular secondary structures are having irregular in nature. Then they are having tight turns, random coils, bulges, some special type of that we call it a super secondary structure also. So here what you have to do is, here you have to identify the the secondary structure you have to predict. So alpha helical structure, I need not tell you, you know that I told you this is the helical like structure. This is the way the helix goes, and you are having the CONH terminal from one amino acid will be interacted with the NH of the CO of the one amino acid will be interacted with the NH of the next side chain. So R1 and to R4. So that is the linkage it will happen. So here you can, and it will fully folded polypeptide chain you will be able to see. Then the beta sheet, you know that this is the parallel chain with the strain. This is the amino acid orients from one direction, from amino terminal to the carboxy terminal end. So here you can see the amino terminal end to the carboxy terminal end. Here it is the just is the opposite direction. So this is the parallel beta sheet, and this another one is anti-parallel beta sheet. Here you can see both the chains are on the same direction. Both of them are in the amino terminal same direction. So this is the parallel beta sheet. So this is the secondary structure, beta sheet. Now another very important when you talk about conventional bioinformatics, especially when you model the protein, the importance of one most important concept, Ramchandran plot. The greatest discovery made by the great Indian scientist, the former professor and great scientist from Indian Institute of Science, Bangalore, and he was a great human and a very great teacher as far as the protein chemistry is concerned. His contributions everybody are saluting actually. And a couple of years, you know that I think everybody are knowing, we have celebrated the 50th year of Ramchandran plot. So Ramchandran, G. and Ramchandran, the great scientist, and he identified the secondary structural the psi and phi angle of a secondary fully functional secondary structural conformations. So and he plotted the psi angle along x-axis and phi angle phi angle along x-axis and psi angle along y-axis, and he got a plot, and you can see that and in this particular plot the degrees of freedoms, that is the peptide, where the peptide, which angle the peptide it is coming in each and every amino acid, that is shown. And this particular it is shown that in the upper top side you can see this is if the amino acids are this Ramachandran plot consisting of 4 regions, in the upper left region, upper right region, lower left and lower right. So any residues are comes under in the upper right left region, that will be, we can assume that it will be this is alpha helical beta pleated structure. This is the sheet actually. Then any structure you can see here somewhere around this particular region, that you can see that they will be a helical like structure, and wherever these regions are there, these are the torsion angle required to produce the secondary structures such as alpha helical structure and beta pleated structure. So the stability or stereochemistry of a model we are going to look by looking this Ramachandran plot, and these areas are known as most allowed region. So these areas are most allowed region, and the other areas there is no dots are known as disallowed region. Whenever we model a particular protein, these particular things we are going to check whether your modeled residues are in agreement with the Ramachandran plot, whether their side chain or the torsion terms are in an agreeable mode, whether they have a very specific beta sheet, then the residue will come here. Then they are having an alpha helical structure, then residue will come here. Right hand alpha helical structure, left hand alpha helical structure, both the things we can see there how their torsion terms will be coming. So this is another very important contribution in which we should proud of that one, being an Indian, the contribution an Indian scientist towards protein chemistry. And the next one, the structural classification, you are having the structural classification, lot of structural classification of proteins are there in terms of advanced bioinformatics. Structural classification of protein, that is known as SCOP. Then similarly class, architecture, topology, homology, you are having CAT. Then you are having a family of structurally similar protein or FSSP. Then you are having protein classification or P class. Then you are having CAS, critical assessment of protein structure prediction. Then you are having a dictionary of secondary structure of prediction, and many more. These are some of the examples of databases. They classify proteins based on the specific type of secondary structures. Now this I have already told you. They are having this is just an example of a DSSP classification. How it will classify H alpha helical structure, G stands for 3-turn helix, I stands for pi helix, E stands for beta strand, and B stands for beta bridge, and T stands for beta turn, and S stands for bend, and C stands for coil. Similarly, CAS classification, they use different notation to denote. Then here you can see the other database called CAT, class, architecture, topology, homology. In this particular database, the structures are classified in different groups, mainly alpha or mainly beta. Here I told in the beginning, the first level, the mainly alpha, all structure that consisting of alpha helical structure. Say for example, your hemoglobin, hemoerythrin, okay, all such kind of structure you can see mainly alpha. Then mainly beta, you can see this particular structure is mainly beta and forms a beautiful barrel here, barrel over here. And similarly, most of the structure you can see they are having both alpha helical structure and beta structure, and some they are in the form of a barrel, some will be in the form of a sandwich, some will be in the roll. These are the specific type of fold in which the proteins are going to form. So that is the class. So the class levels are this one. Then the architecture is nothing but that is the fold. The overall fold is known as the architecture. The topology, that is the super family, is known as the topology, and the homology is the family of the protein. What are homologous protein? And this homology is very important, because homologous proteins are the protein that arises from a common ancestor. Such a kind of a protein is known as homologous proteins. Then the fold classifications are also there. As I told you, the fold is mainly all alpha or all beta with very little alpha helical structure, then alpha and beta. This is classified, this particular structure is known as SCOP, structural classification of proteins. Then why it is secondary structure prediction is very, very important. Before we go to the tertiary structure prediction or modeling, you should have a good secondary structure. I told you, fully functional protein means their secondary structural elements play very, very vital role. That is why. And it was so much complicated problem. So many years people have been working, nearly 40 years the history of biological science people have been working, and protein identifying the secondary structures was a very critical task. That is what the crystallization they are doing, actually getting the identifying the probable structures, that is alpha helical structure, beta pleated structure, etc., etc. And you can the structural changes and the comparison of the structure, the structural changes. If these two structures are structurally similar, if you compare these two structures, you can see that they are expected to perform same function. So that is the importance of this particular concept. Then the prediction methods for secondary structure also in terms of computational biology or bioinformatics. First one is the statistical methods. Then you are having the nearest neighbor method. Then you are having neural network method. Then you are having support vector machine method. Then you are having advanced aspect using the machine learning as well as the deep learning approaches. You can see one example is hidden Markov model, that is a probabilistic and statistical model. And the statistical method, the first one is Chow-Fasman method. You are having under this statistical method, and this is Garnier, Oxtrup and Robson, Gore. And you are having another tool called SOPMA, self-optimized method with the prediction, self-optimized method and alignment and predict protein and proteins forecasting secondary structure and JUFU. Then similarly SSPAL and PhD Sec and CyPret, JPret, NN Predict and HNN, that is neural network, okay, hierarchical neural network. So lot of computational analysis it is required. These are the major type of prediction method. Now I will show you a couple of examples how you will be able to do a secondary structure prediction. So this is Chow-Fasman. How I will be getting this page? Simply you go to Expasy under secondary structure prediction. Under that you can see more than 25-30 different tools. Whatever I have told, that is very limited number. So many tools you can see. Under each and every things you can see this different tools, and there you can see the tools. And one such tool is Chow-Fasman. Simply just you click, there will be a box, you paste your sequence and you have done with your work actually. Or you can simply enter your the accession number. So this is the FASTA format, and simply click this button called predict. Then what you will be getting, this is the result is very, very, very fascinating. Here you can see the algorithm, powerful algorithm 10-10 set, the sequence of converted in 10-10 set under, and there is a graphical representation, and this red indicate these are the region they have alpha helical structure, and this green indicate these are the region they are having a beta sheet, and this blue indicate these are the region they are having turn. I don't know whether you are able to see, there is a yellow, these are the region they are having the coil. Okay, here you can see, below you can see this is the Fourier sequence, this is the sequence. It is written in the first line 70 character. Under each amino acid which probable secondary structures will be produced. Some amino acid, here is a serine that can produce both helices and sheet, and which contrast depending upon the functional aspect, the folding when it is performing, that will acquire the ideal secondary structure. So that is what is the importance of secondary structure prediction. So finally to optimize the secondary structure predicted, this is you can see this M that is predicted H, then the V the valine, that is this is again helice. Then here you can see K, that is lysine, that is again helice. Then similarly serine, that serine you can see here it is E, that is extended beta sheet, and so on until you will be able to completely you will be able to get the complete structure of the secondary structure you will be able to get. Then you are having another excellent method that is called self-optimized methods and alignment SOPMA, and this SOPMA is the popular secondary structure prediction tool available in the Expasy server. And here also there is a box, just to paste your sequence and you can click the submit button within a couple of seconds. And one important thing, when you are doing the previous case, in the case of this one, you can retain this FASTA format symbol over here. Okay, you can see the greater than symbol and you can see here, but in the case of here you need to remove the FASTA, the header information. That FASTA format along with one line will be there. So it will, if you include that one, you will end up with certain error, and the computer will not identify, and you will be able to end with certain error. So that you need to remove while doing this exercise. Only amino acids, starting with, you know that amino acids, all the starting amino acid will be methionine, valine, lysine, okay, serine, leucine, arginine, okay, glutamic acid, and so on. You will be able to see. Then this is the same result, and here it will be predicting. You will be having all the secondary as well as super secondary structure. It will be predicted. Okay, so here Python helices, beta helix, beta sheet, and extended strand, bend region, random coil, and everything it will be predicted actually. So this is the output, and here you can see a graphical interpretation also. By looking, you will be able to see what is the predominant structure, whether it is a helical like structure, whether it is a sheet, whether it is a turn, and you will be able to see the result. Okay, then like that you will be having Cypret. This is the beautiful server maintained by the UCL Department of Computer Science, Bioinformatics groups. Okay, and here also you can input the same sequence, and what you can see, same result you will be getting. Here you can see more a colorful output you will be getting. The pipe-like structure, this is indicating the helices, and you can see the arrow mark, you can see that is indicating the beta sheet, and you can see the below you can see wherever the alpha helical structure, beta sheets are there in that amino acid, it is clearly predicted actually. So that is very, very important. And one important thing, the bioinformatics doing it is very easy. It will take only 1 or 2 seconds to complete it actually. But analysis and interpretation of the data, it is very, very important actually. Analysis and interpretation. People should know that when you are getting an output, how will you interpret that output? You should be in a position to understand. So that is very, very important. Then till now, all the previous tool, the input was a sequence. Okay. Say for example, you are having a structure. So what will you do? Is it possible to predict the secondary structure? Say for example, you are having a crystal structure or a coordinate file, or you are having your own a model. So what do you have to do? You wanted to predict the secondary structure. Then what do you need to do? You can simply paste, you can browse and upload the secondary the structure over here, and simply just you click these visuals, and you will be getting a beautiful output like this. Here you can see individual amino acid. Then there is a box, you can see when you cross over your mouse, it will change the psi and phi angle of every amino acid. It will display here. Okay, since it is a screenshot, it is not showing that one. So below you can see the amino acid, and here you can see this red indicate wherever there is a red, that is a alpha helical structure. Wherever there is this green, that is extended beta sheet. Wherever there is this orange yellow, that is a beta bridge. There is a pink color that is not there here, that is a beta bridge, and the coil, this is a yellow color. So like that, you will, by simple visual interpretation, you will be able to see the result. So secondary structure is already over. Now we are going to go for the core concept of our talk. How will you model a particular protein? That is known as tertiary structure prediction, the three-dimensional modeling of the protein. So I told you this is the most important method, identification of molecular target. So why it is very, very important? Here it is very, very complex. Experimentally, I told you this facility, the extracrystallographic facilities are known as synchrotron. This synchrotron is shown in France and so many other places. There are very limited number of synchrotrons, and very well equipped synchrotrons are in the world. Similarly, this is 750 MHz NMR facilities. There are very less number of laboratories will be having this much of high-end NMR facilities. Okay, so it will be because of all this problem with the crystallization or NMR structure. It's a very complicated process actually, and that is why here we are using the model. Now I told you as of now, if you take the system, the interaction studies, so many proteins, there are so many proteins are there, billions of proteins, trillions of proteins are there, and their amino acid sequence it is possible to study, because the massive development in the sequencing approach, all the sequence are available, but not structures. Structures are very complicated because all these things are very difficult to get it actually. If here the graph over here, this shows the here along x-axis, and the number of sequence versus the structure. The UniProt, one of the most popular sequence database where the amino acid sequence hosted, the growth is like this. You 2010 and 2020 it will go very rapidly. Now you are having the next generation sequencing approaches. So because of that one, the amino acid, the nucleotide sequence will be so many that we can convert into protein sequence. So many sequence will be there, but you see the corresponding structure. It is very slowly going actually. Now you see 2010, you can see it is like this, and now 2020, there is a huge gap between the number of sequence and the structure. What I mean to say is so many sequence that doesn't have any three-dimensional structure. That is what we are going to model and we are going to study. We are going to predict it actually. And as of now, and this 20, as of the recently, this is what is the screenshot of the protein data bank, conventional experimental crystal structure. It will show. I told you as of yesterday, 25th May 2020. If you check this sequence, I think this is an approximate number. Crores or millions of sequences are available in the database where amino acid sequence are available. But in the case of structure, you see here clearly, you can see here 1,64,174. This number will change. I have taken this data as of yesterday, 25th May 2020. And you can see, I told you about a beautiful bioinformatics server. This you see COVID-19 coronavirus research. Which all are the new structures are identifying. I told you one beginning. I showed one paper, Nature paper. They have crystallized the main protease that deposited here actually. So that is whatever it is happening regarding the COVID-19. If you open this particular area, you can see that up-to-date information about the research. That is what the role of computational biology. No other science will give you such a kind of a latest development in the field of science and technology. So that is the huge difference. So I told you 95% of the proteins, still the three-dimensional structures are not available. That is the problem over here. So that is the problem here. Then what you do? It is very essential to do the three-dimensional structure. Why? Because I told you the significance of three-dimensional structure. This is the primary sequence data, and they fall in a very specific three-dimensional structural aspects, and they forms complex network. Then this complex network are the functional compartment of the body. If you check any networks, glycolysis, Krebs cycle, citric, any glycosylate pathway, Calvin cycle, any pathway, just to take it, or amino acid metabolism, carbohydrate metabolism, nucleic acid synthesis, okay, any organism, complex metabolic pathway we can see, and individual protein that plays a major role, okay. And this network is a very complex network. That is why one important area called systems biology, it is very important. And this complex protein will interact within the form of network, and they interact protein interaction, and they perform biological function. That is the way the biological functions are happening. But the question is, as I said, but the available structure is only this many structures. Also, we are in 2020. Only we are having 1.6 lakh structure. 95 to 98% of the structure still unknown. Nobody knows. That is what the power of computational biology we can model. So this is the network as I studied. This is from one of my paper, and this is the network, that is a metabolic aspect of a network of we have created by using string database of Salmonella typhi, a gram-negative bacteria that causes enteric fever. You know that, and it is one network, functional network. So individually you can see these are proteins. These particular proteins are connected by the networks, and they are having that is known as nodes, and these are interconnected, and individual proteins can be act as the target. So if you block one particular protein by using a small molecule, then what will happen? It will stop expressing that particular pathway, or it will partially block the expression of other pathway, or it will stop the interaction between other proteins, so the metabolism will cease. That is the fundamental concept of drug discovery. Okay, so what we are going to do, identifying the metabolic pathway That is why I told systems biology. It talks about the metabolic system. It is very, very important. So this metabolic pathway, by looking this metabolic pathway, you can see so many unique proteins, and that particular protein you should identify and check 3D structures are available. If it is available, well and good, you can take. And if it is not available, what you do? Predict the structure by computationally. So that is what we are going to see. So what are the major important approaches for the structure prediction? There are three important approaches for the structure prediction. One is homology modeling. Next one is fold recognition or threading. Next one is ab initio modeling. Okay. So homology modeling, as the name indicates, is otherwise known as knowledge based modeling or comparative modeling, and this is a sequence that is available. That particular sequence doesn't have a three-dimensional structure, and we are comparing that particular sequence with another sequence that has a structure. Okay, and by comparing with a non-sequence that has a 3D structure is the basics of homology modeling. So that is known as homology modeling. And what do you mean by fold recognition or threading? It requires a similar structure, a small region. It requires a particular fold, a particular topology, a particular fold. So if you are able to identify a particular fold by comparing it with a fold library, you will be able to predict an unknown fold, okay, by looking the similarity of the sequence. The third one is, the first two methods are excellent methods. The third one is ab initio method. So there are, when we have to use comparative modeling, when we have to use threading, when we have to use ab initio, there are certain cases. Okay, so if there is, we cannot predict the structure by first two methods. There is no way we can predict the structure. There is no informations are available. There is no threading, similar folds are available. There is no homologous protein that has an experimental structures are available. Then what will you do? You are going to predict the three-dimensional structure of a protein using from the basic amino acid. So that is known as ab initio or de novo from the first principle. It is known as first principle. This is the major approaches. Now we are going to discuss all those concepts in detail. What do you mean by homology modeling? So as I said, it is a construction or a generation or creation of a high resolution of an atomic model that doesn't have a three-dimensional structure from where basic amino acid. That is the power of computational biology. Here you require only the basic amino acid. See, imagine a simple amino acid sequence computer program can create a fully functional protein. If somebody crystallizes later, that is very identical to that particular protein. That is what people have been working so many years. That is what we could not do. Computational program can do. So that is the thing. And here you can see the protein target that doesn't have the structure, that is known as the target. I told you why it is known as homology modeling, because it checks the homologous relationship. Here the one important partner is known as template. Without a template you cannot do a homology modeling. What do you mean by template? The template is a reference. That means to say a target sequence is there. In their individual amino acid they have either identity or similarity. Similarity means their relationship in their physiochemical properties of the amino acid. Identity means it is exact match. In their sequence alignment, if you align the sequence one below the another, so they will be having high amount of relationship, and the template will be having a structure also. Okay, then what will happen? We will take that particular structure from RCSB, PDB or structural databases, and we will make the 3D structure of an unknown target by comparing it with a non-homologous structure. Thus it is known as homology modeling. Then what are the steps of homology modeling? The main important steps of homology modeling are, that is nearly, there are so many steps are there, but mainly they are target identification, the selection of the template, the sequence retrieval, model building, side chain optimization, building the loops, and refinement of the model, and finally evaluate the model. Okay, so this is the all the steps. Okay, this we will discuss in detail. Then this is the overview. As I said, you have to start, identify the related structure. This is what is your target sequence, doesn't have any crystal structure. The other side you are having a template structure that is already having a crystal structure. What is the speciality? This sequence and the sequence of this structure has high homologous relationship, and what will happen? They will do a pairwise alignment. There is a something called as alignment in bioinformatics, pairwise alignment. Here you can see pairwise alignment, and if they are having homologous relationship, and what will happen? Once they are doing the pairwise alignment, which are the area they are having high homologous relationship, the program, the computational biology program will take the structure, grab the structure from the template and build the model. So finally they will be checked for the good quality, and finally they will be getting the structure. So that is what, identify the related template. As I said, select the template, then align the target and the template, then build the initial model and evaluate the quality. If the quality is okay, then fine, end. If it is not okay, then go back to the initial stage, and the problem might be due to the selection of the template. You might have not selected the appropriate template. Selection of the template is very important in the case of homology modeling. So selection of the template, then this particular process you will do. Once you are getting a good model, you will be ending the process. Now the first step, target identification. So target identification means what? You are identifying the a particular sequence or the research sequence that is obtained from a database, either from a database or from the sequence in your laboratory. You might have sequence, or a small peptide you might have sequence, or a gene might have sequence. That is anyone you can use actually. And here you can see in the case of a database, this is a GenBank database, NIH server. You can see again they are providing a lot of valuable information about COVID-19. You can see here the message over here, and next to another beautiful, this is a nucleotide sequence database, and similarly you are having a DDBJ EMBL, that is a European nuclear as a ENA. Okay, then you are having EMBL also. Then, but this is a protein sequence database. In this particular database, you can retrieve nucleotides, and but protein also you can retrieve, and here you can retrieve the specific protein. And again you can see COVID-19. All as of now, if you look, you can see a particular results of 46 new COVID-19 sequence they have deposited. I don't have that particular data right now, but you can see, open this UniProt database. This is one of the best. Somebody asking you what is the best database in bioinformatics, the answer is UniProt. Okay, this is highly curated, annotated database. It has a detailed information of each and every entry of the protein. So UniProt database. From any of this particular database you can select the target. So what is the target? The protein sequence in which the structure is not available, that is known as a target. Now that you wanted to model. Then comes the selection of the best template. So this is the most critical step. How will you select the template? The selection of the template can be done by using similarity searching. Similarity searching, you might have heard about basic local alignment search tool, or BLAST and FASTA. Okay, the BLAST and FASTA are the best method for similarity searching, and you can use, I told you, similarity searching against what? Against the structure. So what you are going to look here? Here you are going to look the structure that has a high homologous relationship with your target sequence. So that structure can be deposited, PDB, protein data bank, then molecular modeling database, MMDB, PDB-SUM, protein data bank summation, PDB literature, then class architecture, topology and homology. I showed you the classification. Then scope, structural classification of protein. You are having interpro. Similarly, so many tools. And how you are going to select the best homologous template. The homologous templates can be selected by the basic local alignment search tool. Now you are having so many BLAST. It's one of the excellent tool developed by Dr. Stephen Achul in 1995, and that is published in the Journal of Molecular Biology, and that one of the excellent tool that you can search similarity. Any similarity with any sequence, it will be you can search, and you are having so many algorithms in BLAST. P-BLAST, if you use a protein sequence, you can use a protein BLAST, and similarly you are having nucleotide sequence, you can use N-BLAST. Okay, here you can see special type of blast called precision specific iterative blast or PSI blast, pattern hit initiated blast or PHI blast, delta blast, that is domain enhanced lookup time accelerated blast, okay, a smart blast, phylloblast, primer blast, so many types of blast you can see. Then certain specific type of region called a motif. In I told you about domain, the functional unit of a domain, that is a small structures of an amino acid that has highly conserved structural region, that is known as a motif or a pattern or a signature. The level we are coding, that is known as pattern or regular expression. How we are writing a particular motif, that is known as pattern. We are having Mimi tools or must, all the tool you can use. And next tool you have to use multiple sequence alignment tool. Okay, so you wanted to align your sequence. So say for example you wanted to select generally we will use one target and one template, but you can use multiple template also. So if you retrieve multiple templates, say for example multiple template, then you wanted to know which template you will select for modeling. In that particular case, you should do multiple sequence analysis. So then you will be having the data, and finally you will be getting the phylogenetic analysis. You will analyze the evolutionary relationship. Okay, so you will be analyzing the evolutionary relationship. You will be getting the result. So, and what are the important parameter or factors one should look while selecting the best homologous template? So the most importantly, one should look the alignment score. So the alignment score you should look from the BLAST result. So the alignment score between your target and template, it should be always high. High alignment score will give you high result, good result. And next one, E value, that is expected value. That is a statistical parameter that is using to understand the occurrence, okay, that is by expecting by chance, there is statistical value that is expressing in basic local alignment search tool BLAST, that is E value, but E value should be lower. So the higher the E value, similarity or identity by homologous relationship will be very poor. Lower the E value, it will be very less. If you are having a negative E value, you will be getting a good result. If you are having a positive value, quality will be very poor. Then next one is the percentage of identity. So whenever you are selecting a protein, should select a protein with a minimum 30% of identity. So 30% of identity is the minimum cutoff you should take it for a protein that you wanted to model by using homology modeling. Why? Because 30% of identity range is the one that will call it as a homologous protein, the protein which arises from a common ancestor. So the percentage of similarity and maximum, what is the maximum? Till it can go to 99. When the percentage of identity increases, the quality of the model of your target, you will be able to get a accurate quality model. When the percentage of identity increases, the quality of your theoretical model will be increases. Minimum is 30, maximum is till it can reach 99. If anybody is able to get a sequence of 99% of identity with your query protein, then you will be having almost a similar structure with that query protein. Actually, there is no highly accurate structure. So depending upon that particular things, you can look. Next one is the query coverage. The one of the size, okay, almost similar size. One particular protein is having 50 amino acid in length. Next protein, whatever is present in the database, has 100 amino acid, but it shows some good E value and alignment score. And if you take that particular thing, you cannot model properly because it has only your target, that particular protein has only 50. Okay, your target has what? 100 amino acid. Then 50 you cannot model. So that is what. Then you are having a percentage of similarity, then length of the sequence, then the resolution factor of the template, highest resolution, that is the highest resolution will give you poor quality, lower resolution, resolution close to 0 is always give you a good quality result. Okay, that is the good quality result you will be getting. Then the environment, the similarity, you know that crystallization is happening in a solvent system, the importance of pH, the importance of a ligand, quaternary interactions, etc., some metal content, etc., etc. So all those things you should be in a position to look here. So selection of the best template, you will be having so many parameters. So this is about multiple sequence alignment. Here you are having the first one is the target, and the second, third and fourth one is the template, assume. And among this one, which one you will select? You are having a confusion. Then you do a multiple sequence alignment. This is the output. You can see the star indicate highly conserved region. You can see the amino acids are arranged same amino acid in all the region during the point of evolution. Okay, it doesn't change. There is no mutation at all. It doesn't allow the mutation. That is highly conserved. That is what I told. Here you can see a common motif over here. Okay, here also you can see a common functional motif. Then which one you will select as the best template? Here you can see first two, these two are the phylogenetic tree. You can see multiple sequence alignment. Later you will be getting a phylogenetic tree, and the tree, from the tree you will be able to get like this, and you can see these two sequence. Say for example, this is your target, and if this is your template, undoubtedly you can select this one. Why? Because your target and templates arises from a common ancestor. This has high amount of relationship with the your target than these two sequence. So you will select that sequence for modeling. Then what will you do? You will align the structure, the three-dimensional structural alignment, the backbone of the target. If alignment is okay, then what will happen? The program will create an initial model. That program you are using, the program you are going to an initial model, and that particular program will create an initial model. And what will happen? That you will be able to thread it or superimpose the model, structural alignment. So one is your target, another one is your template. You wanted to know wherever there is a variation, your target and templates are perfect fitting is happening or matching is happening, and this matching relation is known as root mean square deviation. In root mean square deviation, you can see the root mean square deviation is 0, means 100% of identical structure. If root mean square deviation is 0 to 1.5, you will be having high. There is a very less relationship. Here you can see most of the regions are almost aligning. You can see there are slightly some variation in the regions. Okay, so one is the target, one is the template. So what will you do? How to do it? This one by using superimposition technique, and this is a C alpha traces calculation. The position of the C alpha atom you are tracing. So that is known as root mean square deviation. And the backbone RMSD. Obviously you can see any structure less than 2 RMSD you can consider as a good quality structure. 0, closer to 0 RMSD value is always good. Okay, 2 to 3 it is almost okay, not good, but more than 3 it cannot be considered. Okay, and what a bioinformatics tool you will use for the superimposition. You are having so many tools like Aladdin, Dali-Light, SysPDB viewer, Superpose, ProsUp, then similarly so many other tools you are having actually. So once you do a structural alignment, then what you see, if you are getting this much things, your model is ready. What you have to do? Your program, you tell your program to just hide the template, then what you are going to get is your model. That is what is the model. So this is the RMSD, the model accuracy. The first case, I'll show you this high accuracy. You can see the yellow is your the target, and you can see the red color it is your it is your template. Okay, you can see 100% matching RMSD value. You can see 0.41 angstrom, high accurate, excellent structure. There is a high amount of relationship. But there is a second one, you see there is a almost a similar result. Okay, but you can see there is some variation. Okay, but there is some variation. Okay, identity percentage. First case identity percentage, you see 77% of identity of the template. Then here it is 41% of identity. Third case you can see that is there are a lot of variation. There are some of the region there are no this thing at all. Some of the region there are no region at all. You can see there is not template at all. So many variations are, these are not superimposing properly. So the structures are in different structure. So that is why you are getting a low accuracy structure. Then you may ask, then why it is better RMSD value here? But whatever it is superimposed, that is a perfect superimposition is there than this one. Okay, so that is why there is a slight increase over here, 1.34, here it is 1.17. okay, but still this is better because this covers maximum part of the structure. Okay, 41% of identity is also there. Then what is the model building principle? I told you the amino acids are aligned. Once you are getting this much result, just to hide the target, then you just to hide the template, then you will be getting the model. Okay, so every step atoms, each and every amino acid placed in a proper position. There should not have steric clashes, there should not have problems, or amino acid coordinate should be perfect. They should have a very less strain between this amino acid. That means to say they should be, the energy should be very less. Okay, and these are some of the model building programs. Okay, how will you do a homology modeling? These are some of the tool you can use. Initial model building program. The model building programs are, you can see this is a freeware, Swiss PDV viewer. Okay, that is a free software. Swiss model, that is a web version. That's an excellent tool. Then 3D jigsaw, then EC Pred 3, ITASR, then CPH model, then modeler. This is one of the excellent tool. Then 3D jury, then Yasara, then Discovery Studio. So many versions are available. So many commercial softwares are available, and you will be able to use this particular tools for modeling. And how will you use visualization? How will you visualize this? So these are the most important. I have shown so many images. Okay, so many images I have created. I have shown this images. This is how it was created by using beautiful visualization tools. We are having so many visualization tools: raster molecule, rasmol, PyMol, grasp, JMol, molmol, KIME, visual molecular dynamics or VMD, web lab viewer light, okay, UCSF camera, ACD 3D viewer, hex. So many software, modeling software you can it is available for free of cost or commercially available. Now the next one is, now you have made the model. Now the next thing is side chain optimization and loop modeling. This is some advanced aspect of molecular modeling. Generally it will be very difficult to do a normal modeling. So what I wanted to tell you is, here you can see in the initial alignment, the first line there is an alignment. This is what is the target. Okay, and what is happening? This is the target. Then second one is the, first one is the target, and first one is the template, second one is the target. And you can see while it is aligning, then this particular in order to make excellent quality alignment, the program introduce certain gaps, so that you can see that this particular, when modeling, when making an alignment, V and V is matching, this is matching, this is matching, this is matching, all those things, three-dimensional structures are available. But here, see this region, there is no 3D structures are available. So what will you do? The program will not model that particular region. That we can see. All other region, here also it is there. With this particular region, what it will, the program will do? Program will create a loop. Okay, so that is lack of template over there. Then what you need to do? You select that particular loop. There are loop libraries. Again, you compare with certain loop libraries and make that loop. And that small, now you see there is a loop. Using this particular small peptide, you have created a loop. Using this loop, you have created a peptide. Finally, by using that loop library, you need to stitch it onto this particular structure. And what you need to do? You can fill this particular region over here. Okay, so you can then complete the model, and this particular model is known as loop building. Advanced software only will be doing this particular step. The next one, the model refinement and validation. Once you have made the model, you need to prove that your model is a good quality model. How will you prove that? You need to make sure that your model should be useful for the main research. How will you do that? Somebody crystallizes or somebody identify that model. If they get a model and their model and your model should not have that much difference, then only your model will be appreciated. Otherwise they will not appreciate. Okay, how will you qualify that particular appreciation? How will you do that particular modeling? The main things are known as energy minimization. So that means to say, I told you, when problem is when the protein is not having the stability, when their secondary structural alignments are not proper. Okay, the secondary structural alignments are not proper, what will happen is then that particular case, the protein will be showing high amount of energy. So then you need to minimize the steric clashes, and as I said in the beginning, when protein folding, many conformations will be there. Which conformation will be giving the functional properties? The bioactive conformation, that particular property. How will you do that? In order to mimic that particular process, what is happening in the protein folding, we are doing energy minimization. We have to minimize the energy, and you have to get the best, lowest energy structure. You will be getting n number of model, and which is the lowest energy structure in kilocalorie per mole. Okay, so that you will be taking as the best model. And another thing is you can consider Z score, and there are some cutoff for the model quality in terms of Z score. And what are the bioinformatics tools you are going to use for the model refinement and validation? There are two tools. There are a lot of tools. Okay, one is known as ANOLIA, Atomic Non-Local Environment Assessment Evaluation. Okay, GROMOS, then GROMAX, QMEAN, Verify 3D, ProCheck. This will create this will check the quality, stereochemical quality, especially Ramchandran plot. Then WATCHECK, it will check many stereochemical properties. Okay, then Verify 3D, it will check the error, the atomic compatibility, 1 to 3D ratio. 1 means that is a sequence level, 3D means that is a structure level. That compatibility it will check. Then you will be having ANOLIA, that the potential energy will be calculated. Okay, you are having vadar, volume, area, dihedral angle reporter. As I told you, the quality of the peptide unit it will check. Then prove, it will check the statistical Z score. Previously I have spoken. Then PROSA, protein structure analysis. This will compare the Z score of the experimentally solved structure and with that the Z score of your hypothetical model. Then the dictionary of secondary structure of protein, DSSP, it will check the secondary structure. Then similarly ERAT, is there any non-bonded interaction is there in your protein, that will check. Then CHAMP, that is energy minimization. ProMotif, you will be able to, they will see if there any specific type of a motif. Okay, then you can see this is what is the first step, the ProCheck. So if you analyze your model, these two structure I have shown. This is what is this? I told you the Ramchandran plot. That is what I have initially I told about Ramchandran plot. Now you are going to analyze the protein in terms of the stereochemical. Your secondary structure, the psi and phi angles are whether they are in a perfectible agreement or not. So here you can see the red region. You can see this region, as I said in the beginning. You can see this is the upper left side of Ramchandran plot. This is upper right side. This is lower left side and lower right side. And this region over, that is the color. This region color that is shown in red color, that is the most allowed region. This region, and this one this is additionally allowed region. This better. Okay, this region, that means to say every amino acid should come. This particular region is excellent. Then quality secondary structure is perfect. Then if this particular area, it is okay, no problem, it will perform function. But this particular region, it is not that much good, but it is allowed. This is known as allowed region, and these wide regions are non-allowed regions. Okay, so what you have to do, you have to check the quality of the protein, whether your protein residue secondary structures are in agreement with the Ramchandran plot. Individual that dot represent one amino acid. Okay, if you click, you will be able to get the psi and phi angle of that particular amino acid. Here I told you, here you can see this is a beta sheet, this is a alpha helical structure, this is again some alpha helical structure. This particular plot, you see none of the amino acid residues are in the disallowed region of the Ramchandran plot. Thus this structure is a good quality structure. Okay, there is no structure you can see in the disallowed region, no protein, no amino acid sequence. Okay, however here you can see over here most of the residues are in the white region. So hardly you can see amino acids over here, but here you can see most of the amino acids are the white region. So obviously the energy, a lot of energy clashes, secondary structures will be not proper, it will be a problem. So what will happen? It will not be giving a good quality structure. Okay, so that is the speciality here. So that is the importance of Ramchandran plot. Then ERAT, here you can see non-bonded atom. This is the cutoff here, where this is the individual amino acid is given in the x-axis, and there is a 95% of error region, it is shown in the y-axis, and here you can see this particular amino acid, they are having some non-bonded interaction, and yellow maximum green region should be there to appreciate the quality of the structure, but there are some problem due to the steric clashes. So this is 99% of error. Okay, 95 it is warning. Okay, it should come, most of your structure should come within this particular region if you wanted to tell this structure is a good quality structure. The quality here, it is 77.1 particular protein I have tested, it is 77.59, not bad. Okay, the next one, as I told you, verify 3D, it will check the compatibility between 3D to 1D. So when it is converted into a 3D structure, you know that when it is a tryptophan, what type of structure it will produce, valine, what type of structure it will produce. There should be an agreement between a 3D to from the amino acid level to the structural level when it is come, whether it is should have a high agreement. It should pass, you will be getting a message called pass. Okay, so if it pass, then only you will be able to get that. Means to say at least 80% of the amino acid should have a greater than 0.2 in their 3D bar 1D profile. Then only you can call it as this thing as a pass actually. Then that is verified 3D. Next one, gromos, aneolia, cumene, DSSP. These everything are a particular force field. Gromos means Groningen molecular simulation package. Aneolia, I told you, atomic non-local environment assessment evaluation. Cumene, dictionary of secondary structural protein. All those things here you can see. This particular protein responds to aneolia. This is cumene. This is gromos. So what is over here in the x-axis? You can see the amino acids are shown in x-axis. Individual amino acid, what is their peak? This N. Okay, so asparagine stands for what is this? A green peak. That is, energy is minimum, the potential energy is minimum, the stability is lesser, stability is very good, so that is not a problem. Okay, individual, and it is producing an beta sheet. And next amino acid, valine. Okay, it is also not a problem. So like that, from one end to another end, you can see this peak should be maximum, should be in the lower region. This negative potential should be there. Then only you can see that the quality of the protein is good quality protein. And some of the regions there are red, but later you need to refine it and you need to modify and you need to identify the problems over the particular protein. Then so you can see that most of the regions should be, the peak should be towards down, the negative value, the negative potential, so that the protein is a good quality protein. So until from one end to another end it will check, and this is known as the protein. And when you are referring the international research papers, some manuscript, you will be able to see all these pictures very commonly in the figure. So you should know how to analyze that. Doing this one, I told you, it will not take a lot of time. Okay, and I told you about all these three things. Okay, pro check, this cumene, verify 3D, pro check, what check, all those things together. Now a very important server called structure analysis and verification server, or SAS. Okay, and all the tool you need not how to, you may ask, how to retrieve the tool. The answer is very simple. Just you type it in Google, that's all. Okay, you will get the tool immediately ready there. Okay, some tools are hosted in some server like Xpasi, I told you. Okay, so these are freely available tools. Then next one, another thing is like PROSA. This is one of the important tool. Here you can see in the x-axis the number of residues is shown of the modeled protein, and the y-axis it is shown a possible z-score. Okay, and there are two regions. You can see a dark blue color and light blue color. The dark blue color is this is the range of z-score of an NMR structure. Then similarly, light blue is indicating this is the range of the structure in the case of an X-ray structure. Okay, so you can see a dot over here, that is the z-score of your model. Just you open PROSA server and submit your model, it will automatically select the best template and it will assign the model. And this dot should not come any outside this coloring region. Outside this coloring region, if it comes, then the quality is not good. Okay, so that is the important thing. So the quality should be, the dot should be here. So that is about the PROSA. Here you can see the outside, when you are looking, there is a cutoff value, it should be in the below cutoff. Okay, there should be negative energy. Then sometimes what will happen is homology modeling, there will be lot of errors by we are doing. What are the potential errors in homology modeling? So there are sometimes what will happen is the side chain modeling we will do. I told you we are separately modeling the side chain. the wrong side chain come and pack. Actually side chain packaging may be wrong. Then distortion and shift, that will be because of lack of template, it will be a mismatch will happen in the alignment. Sometimes there will not be any template, okay, there is no template at all, then it will produce a long loop. So that sometimes it will happen misalignment. I told you alignment is the critical parameter. See, instead of aligning in the one by one below the another, these amino acids are aligned in a different way. Here you can see there is a variation in the alignment. This is a misalignment, and there is a similarly incorrect template. I told you that's what the most important step. I told you, selection of the template. Template selection is not good, then we will not be able to get a good quality protein. Okay, so this is the incorrect model. Then next one, finally you will do the energy minimization. The good quality protein means there should be minimum energy. What may be this overall energy? That is a potential energy associated with the system of atom. That means to say the bonded atom, overall energy of a system, molecular. Little bit of biophysics is required here. Here you can see the energy is nothing but the stretching energy, the bond stretching energy, the bond angle energy, the bond angle, and the torsion. I told you about psi and phi angle. Then the bond non-bonded interactions. Okay, these are the important types of free energies. Okay, and all those energies should be calculated and make sure that there is no problem. All those energies should be negative. Finally, the energy should be negative. Then only the conformational flexibility should be there. Then there is a term called force field. That is energy function we are representing. What do you mean by force field? The potential energy associated with the system. Okay, and you can see, and the most important problems of the especially when proteins are folding, note the major forces or stronger forces, they are creating the bond. The weak forces are playing a very vital role, okay, such as van der Waals forces, covalent bond, hydrogen bond, charges, hydrophobic effect, etc., etc. All these particular forces are very, very important types of forces. This type of forces you will be able to, I minimize this particular by using this particular process. Then there are three important minimization steps. These are very complex mathematics involved in it. I mentioned, and I skip the slide. One is known as the Newton-Raphson method. That's a big unit actually based on the Newtonian motion. Next one is steepest descent method. Next one conjugate gradient method. There are a lot of mathematical derivation formulas. Okay, molecular dynamic approaches. Okay, so thermodynamics are involved in this particular aspects. And how will you minimize the protein? I told you should minimize the protein by using, and you should make sure that compare the energy. What is the energy before your before minimization, or before you are starting doing the model, and what is the after the minimization? That particular tool called Chimera you can use, and similarly mode refiner you can use. Okay, by using this tool you can minimize it, and the minimized structure you can use. And earlier, now question will be, I told about PDB. Okay, so is there any database where we can host our hypothetical model? So for example, I wanted to model PDB. Earlier host the hypothetical model, but later they have not they have removed our hypothetical model. They host only experimental model, that is crystallized NMR, crystallized by X-ray or NMR method or electron microscopic structures. Earlier they used to do a predicted model also. Okay, now they have changed. But if we wanted to host our model, what you can do is, there is a server called Protein Model Database. There is a beautiful paper in nucleic acid research. You can visit. Simply type in Google Protein Model Database and you can submit the model, and you can give all your details, and it is a very big credit. You can consider this thing as a small publication or project, and whenever anybody wanted to give, and you will be getting a unique accession number for that particular project. Okay, with all the authors you can give the name. Okay, and anybody in the world, they can retrieve that particular protein. You are the author for that particular protein, and the better molecular target if you do, you can download from the data. Similarly, one more tool called Mod Base or Mod Archive. Here also you can submit, and this is a little bit advanced tool. And here also the quality of the model, they will check every parameter. They will check, then only they will accept your quality of the model is very good. Then they will host in their server, and anyone can retrieve for their use. So that is the thing, and that you can cite it in the manuscript, and you will be able to use for structure also. Then the second approach, so this much things I have told about homology modeling. Second approach will be threading. That is known as the fault recognition. Fault recognition is a very very simple method. I told you, we are not going to model a complete protein like a homology modeling. We are wanted to study a small peptide fold, a particular fold. So you are having a particular fold, a small region, and you wanted to study that, and what you need to do, you wanted to model the fold in a more accurate way. Now you should compare with a fold library. So that is known as threading. Okay, so threading, this is the methods, very simple. This is what is your target sequence. Okay, here the diagram over here you can see target sequence. Then there is a template library here. Then the target sequence will first, it will go and do a pairwise alignment with this template, and they will see the alignment. They will go and align. They will check here. If it is fine, it's okay. Then second one, this particular template will match with the second protein. Again they will do a pairwise alignment and superimpose, it will happen, and they will check the RMSD, and they will assess the value. Then again the next protein that is present in the library, fold library, the same process will happen. Your target will be interacted, threaded with the protein that is present in the library. So again it will check the RMSD. Then how many proteins are there, entire protein it will check, and it will be, and finally it will predict the, out of this one, which is the best fold, and that is what, this is the best fold. Finally it will create a predicted model. Okay, this is known as a fault recognition. This is highly accurate, excellent method. If you go to this particular frontiers in bioscience, you will be getting the details of this particular paper. So this is the mechanism of fault recognition. This I told, this is what is your target. So you are having that sequence and you are having the template. Okay, we have to build an initial model. So you have built an initial model over here, okay, by computer, and this initial model, by seeing that alignment, you have created an initial model. Then you are having a big fold library, individual structure you compare. Okay, then what you need to, you should get a best model, and finally if there are any problems are there, you should be in a position to rectify it actually. So you should be getting a non-high quality protein. This is known as a fault recognition. Then next one is the threading. De novo prediction or ab initio prediction. Ab initio means first principle. What will happen? This is less accurate. Is there any method? Then there is no method is possible. We are not having any method, either homology modeling or ab initio modeling. There is fault recognition. Nothing is possible. Then what you can do? If you can check if proteins are having any novel fold. This is based on quantum mechanics, and is mainly used on the physiochemical property. By looking the physical and chemical features of individual amino acids. That is what, my dear participants, I told, the protein chemistry is very very important. The protein chemistry and their properties and their the structure, if you are not understanding, it will not be able to model the protein. So that is the thing. So by looking the physiochemical features, you will be able to make a model that is known as ab initio model, ab initio prediction. Then this is the mechanism over here. This is the sequence, okay, and it created a model. And now what we have done is we are having so many we have created so many structures. This is one possibility, next possibility, next possibility, next possibility, all the possibility we have tested, and some selected structure that has almost equal energy we have selected. This is 1, 2, 3, so many is there. Out of some filtering process, we have selected some biophysical filters by using energy and all those things, best 6. Among that best 6, we have simulated, and based on the best, based on the final energy minimization, which is the best structure that will be applicable for the, for the model. So that one we have got it, and finally we have clearly we have shown the model over here. That is known as ab initio method. And which is the server? This is one of the good server that is using ab initio prediction. So what you need to do is simply you should have a username and password to log in here. Especially you require an academic user ID, Yahoo, Gmail, all those things, it will not work here. And you require an academic license ID, okay, or student's license ID, and you can submit, and it will, the result, it will be predicted the model or like this, and it will send it to your mail. And very recently this ITASER, okay, ITASER, the expansion is iterative threading assembly servers, okay. So that is the expansion of ITASER. It's an excellent server made by the University of Michigan. There is Zhang lab, excellent contribution University of Michigan, bioinformatics, one of the very world famous university. They do lot of bioinformatics research work. Then here you can see a new tool. It is launched in 2020, very recently. You know that this is CI ITASR. Now due to COVID-19, they are so many people have submitted their task to ab initio mode because COVID-19 targets. So many people are there. Here you can see from May 19 to June 30th 2000. This will not be available. Recently, couple of days before I modeled one particular protein, I could get a result. Okay, so that is a very new model that is known as CI ITASR. Okay, this is a version of ITASR. Then next one, till now we have done the modeling. Next, how will you do the drug actually? How will you design a drug? So in order to design a drug, you should have so many steps. Okay, first you should show the drug. When I wanted to call it as a drug, should predict the drug likeness, prediction of ADME, prediction of toxicity, and those molecules qualify all those things. You should go for docking. Finally, you do the molecular dynamic simulation. Finally, you calculate the energy. Finally, this thing will go for experimental evidence. Please note that that is what the drug discovery, conventional biology, for this many things in bioinformatics, conventional biology, if you wanted to do it, a person should spend a minimum 7 to 8 years. A computational biologist will be able to do very rapidly with a rapid time. That is why I told lot of people are working on a bioinformatics approach, especially in COVID-19 era. Okay, here this is what the symbol representation, and a beautiful paper is published in one of the publishers called Bandam Science. You can see that particular paper. Here you can see the target, as I said, the library preparation, the receptor selection of the target. If structure is available, you take. Otherwise, we should model whatever we have discussed. Binding, the prediction of the binding site. You should predict the binding site. The pocket you need to predict it. Then finally, you should first you should do all this preliminary this one. Then when you do one step, what will happen? Whatever the potential problem that can happen in the experimental level, we are trying to minimize. Okay, so there are so many statistical molecules. We are going to filter the molecule. Finally, you are going to select a set of molecules that can have ideal drug likeness, ideal pharmacokinetic features. ADME stands for adsorption, distribution, metabolism, excretion, and toxicity. You know that ADMET. So ideal property you should look. Okay, so all these three things are known as computed. This many protocols are known as computer aided virtual screening. If you do this many things, that is known as computer aided virtual screening. So those molecules qualify all those things, then you will go for docking study. First, you will do a drug likeness. Those molecules qualify, you will select it, and say for example 200 molecule you selected, and 90 molecule will rule out when you are doing for the drug likeness. Then the 10 molecule will select, and that 10 molecule will do ADME. When you are doing the 10 molecule, sometimes 7 molecule will go outside your main basket. Okay, then only you are having 2 molecules. Then when you touch the toxicity, probably one may qualify the toxicity, one may eliminate. So that molecule will go for the toxicity and further studies, and you will go for the experimental evaluation. That is the power of computational biology. So many initial screening you can do, and please don't misinterpret that. One important thing I wanted to tell you: computational biology can only give prediction. So then people don't think like, oh, everything it is available, then computational things can be done. Computational biology will make you to reach the last step. That you require a lot of effort, but last one you should prove in experimentally only. Experimental approach is very, very must actually. So finally you will do docking and MD simulation. Then one things, and drug likeness. There are so many filters. Selection of the potential drug molecule. Drug likeness, you are having Lipinski rule, then combinatorial medicinal chemistry like rule, CMC lead like rule, MDDR like rule. Okay, similarly World Drug Index rule, Gauss filter, Egan filter. Then similarly these are drug likeness properties. Then you are having the pharmacokinetic features, adsorption, distribution, toxicity. You are having human intestinal absorption, in vitro, CSC2. This is the adeno-colorectal carcinoma cell lines, MDCK cell lines, skin permeability, plasma binding protein, blood-brain barrier penetration. All those things are statistical model. How it will be doing in our body by using high-end statistical models. The software is having statistical models. You will be able to predict it that, and you can get the good result. And you are having, how will you do this? You are having so many softwares: Admissor, Schrodinger, Drag-Lite2, Swiss ADME, Lead IT, Fluxus, Pre-Admit, Mol-Inspirant. Okay, and for all of you, you can use Admissor. This is Schrodinger package. It is there. Lead IT, Fluxus. This is a commercial one. Then Pre-Admit and Swiss ADME. Swiss ADME in 2007. There is a beautiful paper about Swiss ADME in, that is published by the Scientific Report. That is freely available. You can use that actually. So these are the common filter. Then drug likeness, I will discuss only one important thing, because most of the people, they use Lipinski rule of 5. What do you mean by Lipinski rule of 5? This Lipinski rule of 5 is a set of actually 4 rules, and we are called it as a Lipinski rule of 5. This is made developed, this is proposed by Dr. Christopher Lipinski. Okay, so any molecule, this is the first and foremost filtering. Number one step, if you wanted to call it as a drug means, so the molecule should qualify the following things. Number one, the partition coefficient should be less than 5. The molecular weight should not be more than 500 Dalton. There should not be more than 5 hydrogen bond donor, or less than 10 hydrogen bond acceptor. Then minimum there should be 15 rotatable bonds. For rotatable bonds, this last statement is not, obviously it is not important, but you can use it. Okay, so these 4 concepts are known as Lipinski rule of 5. Okay, this is very important rule actually. So number of rotatable bonds are increases, you will be able to see the flexibility of the ligand will be increases. Then another very important thing is known as pharmacophore. What do you mean by pharmacophore? Prediction of pharmacophore, you will see lot of paper, QSIR modeling and pharmacophore modeling. What do you mean by pharmacophore modeling? So pharmacophore is nothing but, if any molecule, if I wanted to call it as a drug, they will having a chemical structure. The essential framework that is required for a molecule to call it as a drug is known as the pharmacophore. The essential chemical framework, okay, functional group or the skeleton, okay, that required to call a drug is known as a pharmacophore. Okay, that is an ensemble that is responsible for a druggish property of a lead is known as pharmacophore. Here you can see an NH. Here you can see an aromatic ring. Here you can see halogen, fluorine. Okay. Here you can see another aromatic 5-membered ring. There is a 6-membered ring, it's a hydrophobic aromatic. So like that we are check this. If I wanted to talk about pharmacophore modeling, again I need to take 1 hour lecture actually. So that much information are there for pharmacophore modeling or QSIR modeling. That is advanced bioinformatics actually. So this is the next important thing. Then those molecules qualify the drug likeness. Similarly, the adsorption, distribution, metabolism and toxicity. Then the pharmacophore features, that molecule you can select as a lead molecule for docking. So you require 2 things. What is this? One is the receptor or protein. Next one is the ligand, a small molecule. That can be a natural molecule, or that can be a synthesized molecule, or that can be drawn in a structure, and you can optimize into a 2-dimensional structure, and you can visualize in a viewer. Okay, receptor, I told you, from protein-protein interaction network, you identify if crystal structure is available, take it that. If crystal structure is not available, we have already discussed, that is homology modeling. Okay, then molecular docking. What is molecular docking? The study of the receptor ligand interaction by computational method is known as molecular docking. That means to say the fitting of a small molecule in the groove or a pocket of a large molecule, or that is known as a molecular docking. Okay, there are 2 types of docking: protein-protein docking. That means to say the receptor is protein and the ligand is also a protein. Then protein ligand docking, this is the common docking we are going to use for the drug discovery. Okay, so in the first case, if your drug is a peptide, okay, and target is also a protein, then you have to use protein-protein docking. Second one, protein ligand docking. The protein and ligand. As ligand is a small molecule, protein is the large molecule. Okay, here you can see a fitting of the ligand over a group, and it will be, this is the flexibility. Okay, then here you can see this is a protein, and the binding site you need to predict. Okay, that's a ligand, and it will go for a ligand. Okay, it will go and fit into the pocket. Okay, this will mimic, and it will go and search in all the pocket of a particular receptor, and it will select the best pocket, and one particular protein, when we are doing a docking, depending upon the efficiency of the algorithm and the software, it will go and search so many pockets, and the best pocket where the drug will be effectively binding, that particular pocket is the ideal pose. Okay, so that is the ideal pose. That is the substrate enzyme substrate mechanism. Okay, that is a lock and key hypothesis. Okay, and this particular, based on the three-dimensional structure of a protein, we are going to design the structure of the ligand. That is known as the structure based drug discovery. Okay, structure based drug discovery. Then the pose accuracy, we need to predict it. Then there are two important components in the case of molecular docking. One is known as a search algorithm. That means to say the best algorithm you need to search. Next one is the scoring function, and you need to when the molecule is going and binding, there are a lot of free energy calculation you need to make it. I told you everything is working based on the calculation, energy calculations. So this free energy calculation you need to make it actually. So that program will do very rapidly. So many 2 lakh, 3 lakh binding poses the docking software will recognize and bind and identify. So that is what it is going to happen. So then there are two types of docking again. One is known as the rigid body docking, another one is known as flexible body docking. Rigid body docking in the sense the protein kept rigid and the ligand is allowed to bind. But in the case of flexible body docking, this is a common this one, and the ligand is made it into flexible, and both the ligand and receptors are flexible and natural process, and there is a cooperativity between the ligand and receptor for their interaction, and there is a conformational changes over here. That is known as the flexible body docking. Then so many scoring functions are there. One is force field based scoring function, empirical based scoring function, knowledge based scoring function. Some software like doc, autodoc, these all things are docking software. Different types of scoring function, and what is the efficiency of the software, that depending upon the scoring function and the search algorithm they are using. So accurate scoring function and the search algorithm, if a docking software is having, the prediction result will be very, very accurate. Experimentally it will be very easy to prove it actually. So this is about this one. So this is the overview of molecular docking. Recently we had a particular published a chapter, so that is from there I have taken the molecular docking, searching algorithm and scoring function. So different types of process over here, and what are the main important scoring function, force field scoring function, empirical scoring function, knowledge based scoring function, concerns and scoring function, how it will be doing this particular process. So these are the things. Then when you are doing a docking, how to prepare the protein. When you are doing a protein, there are two cases. One is the crystal structure. When the crystal structure is there, what you need to do, when crystallization is happening, what is happening? There are a lot of water molecules will be there. So the molecules will be crystallized along with the water molecules. Some molecules will be crystallized along with the ligand, closed conformations. So when you are preparing the molecule for docking, you need to remove all the water molecules. You need to remove all the water molecules, and what you need to do is then you need to add polar. We need to mimic that same process, the solvent system in the docking this thing also. So you need to add polar hydrogen, then you need to add charge also to that particular hydrogen, protonation. So once it is done, that is already over. These three steps are very, very important for a protein preparation. And how will you prepare ligand? I told you beginning itself, ligand is a small molecule. It will be having a bond length, bond angle, and the flexibility. So the root of the atom, from where it is going to be flexible, and where is the root, and what is the bond, and what type of flexibility you wanted to make it. So like that, how many suppose you wanted to make a flexible bond into non-flexible bond into flexible bond, you can make the root of the atom, from where the base in which that is going to be flexible, all those things you can make it into the docking software. So that is known as ligand preparation. Again, you need to add the charges and all the steps you need to do it actually. So you need to save it in appropriate format. Then lot of molecular docking softwares are there. They are having the doc, autodoc, Vina, 3D doc, patch doc. This is a protein docking server. All other things are ligand, Argus Lab, okay, that's again protein. Then Fluxx, that is a commercial one. Glide, okay, commercial one. Gold, Legend, okay, Linger, 3D Doc, okay, JumDoc, free version. JumDoc is a very free version. Student can learn with JumDoc. Yasara, and one software in IIT Delhi they have developed, that is known as Sanjeevani. Then Hex, okay, Legend, Green, Gram, Ludi. There are many more. So many softwares are there. From if you have, there are a lot of reviews are available. That review, if you look, there are more than 2020. There are more than 200-300 plus docking softwares are available. Then according to this data is quite little bit old data, but still you can see the usage, which is the best docking software. According to the usage, it says that Autodoc is the one of the best docking software. It gives high 25.87% people they have used, identified Autodoc as the best docking software. Then comes Gold, and then comes Glide, and all those things. Whatever it is, I told you, the final quality of the result should be identified in terms of the docking, should be identified in terms of experimental analysis. Then once you are identified the docked complex, how will you judge your result, whether the docked result is good or not? What are the parameters you need to consider? There are so many parameters. Number one, the binding energy. I told you, when so many docking process will be there, the program will be segregated. The best one will be given the first, next, then comes second, then comes third, like that. Autodoc generally gives best 9 confirmation. Autodoc Vina, very recent one, very good tool. Okay, so that you will be getting the minus kilocalorie per mole. So how much ever minus less value you will be getting, that is the best result. Then number of hydrogen bond with that particular ligand and receptor is stabilized. Number of weak interactions, okay, number of weak interactions, and the binding process of, say for example, if the ligand is having a usual structure, the same binding pocket your ligand, the ligand whatever you wanted to study, it is binding. Then you can co-crystallize the structure, the same binding, it is binding. Then the binding site you can study. Weak interaction such as ionic interaction, hydrogen bond, okay, similarly van der Waals interaction, pi interaction, all those weak interaction you need to judge, and number of interacting residues are also you need to count. Then these are the important aspect one can judge the docking result. Okay, so these are the important parameter. And once you do the molecular docking, most of the people they end with the molecular docking, but that is not correct. You need to, before you are going to mimic that particular things in experimental level, you need to show that the whatever you have done is a good quality structure. Okay, good quality, the docked process is the best one. Then what you need to do, you need to do energy minimization, and you need to do molecular dynamic simulation. MD simulation is one of the most important. It's a very time consuming process. Lot of computational facility it is required. Okay, generally people require lot of high-end servers and all those things. So what is happening? The receptor ligand complex, the stability of the complex, they will be checking in a small temperature, then pressure. Okay, but how your body, the system, if the molecules and targets are working inside the body system, they are creating a virtual environment over there, and we are going to simulate the molecule whatever we have done. Okay, by using docking, and we are finding out that whether the result is correct or not. And the RMSD, RMSF, okay, hydrogen bond formation, okay, the torsion profiles, so many data you can generate actually by using molecular dynamic simulation. You will see lot of molecular dynamic RMSD plot in the in the published paper. Then the free energy, after that the free energy you can calculate. There are two important approaches. Again, it is a very complex thermodynamic, molecular dynamic concept, molecular mechanics, Poisson-Boltzmann surface area, or MMPBSA, molecular mechanics generalized bond surface area, MMGBSA. So whatever the method, you are going to calculate the Gibbs free energy, the overall delta G. Here you can see the overall energy is the energy of a bond, energy of electrostatic energy, van der Waals interaction, polar bond, non-polar bond, okay, and all those things that will be subtracted from the total amount of the entropy actually. Okay, so that is the energy. Okay, so these are the important aspects. Then the prediction accuracy, I told you, once the pose is predicted, what is the best pose? I told you, you should get a pose of very less RMSD variation in the variation RMSD between the protein and receptor, ligand and receptor variations are very close, then there is a high accuracy it is there when you are doing the energy minimization. So that is the main important aspects actually. So here you can see this particular paper I have taken from Scientific Report 2015. There are 3 cases. So here you can see the simulation. Here you can, the first case the receptor is going like this and ligand is going like this. There is a huge deviation. This is not a good result. But second, this here you can see the third one, the receptor is going like this and ligand is almost closer and they are going and binding. They are very much closer and going, that is happening. Okay, binding is almost some region it is happening. So this is very close. The RMSD is very good actually. But here you see the RMSD is very good. There is, you can see initially the receptor and ligand, it is a you can see there is the flexibility, the time, nanosecond time, what is happening, the RMSD, it is what is happening here. You can see the RMSD is less than 3. Okay, here you can the variation is very less actually, depending upon the corresponding docked conformation. Also you can see here you can see there is a hydrogen bond. Here there is no proper bond at all. The RMSD is very high. This one also RMSD is going from starting. Initially it was started low, but it is going high. Here also, but it is RMSD should the value should stabilize actually. So this is the docking. So you will be able to see core computational biology papers. You will be able to see molecular dynamics simulation. There are some of the journals without molecular biology, without molecular dynamics simulation, they will not accept. Say for example, one journal called JBST. Okay, Journal of Biomolecular Structure and Dynamics. One of the core concept is they should have a, in the absence of an experimental data, they should have a high-end molecular dynamic studies. Okay, and the molecular dynamics is based on your system requirement, and similarly your software. You can, there are a lot of tools. What are the tools for molecular dynamics? Schrodinger you can use. Okay, they have a Desmond tool. Similarly, NAMD you can use, and most of the people they use a Linux-based platform called Gromax. Okay, so you can do molecular dynamic simulation. Okay, so that is the next important thing. So here you can see a molecule, a protein. This is what I have told. A protein, it is how it is docking. Okay, how it is docking. You can see visually. This is the protein molecule. You can, this is shown, ribbon and side chain is represented as the wire frame, and you can see the binding pocket. Okay, and the molecule. See, I told this is the way docking. Whatever when you are doing, it is happening. It will go and bind in so many pockets. You can see the conformational changes in the ligand. The flexibility of the ligand is changing. You see, this is binding. Start trying to bind. Almost they are locating a portion. They are located. Now you see fine adjustment. They are going to take place. See, they are fine adjustment in terms of energy. Okay, the slightly they will adjust first minus 11 point increasing slightly, -1, minus 11.70 to minus 93, minus 12. Slightly you see the side chains are minus 13, minus 14.11, minus 16. This is the best one. So this is the best result you could get it actually. So this is this is what actually the docking processor will do actually. Okay, so and you can see there are people may ask is there any commercial drugs they are available designed by computationally. Yes, many drugs, even many companies, Merck, Roche. Okay, so you can see this from I have only last 10 years data. Now after that, after 2010 2020 there is a huge one. Now I told you can see that people are working on even all the the SARS. You can see so many drugs actually. They are repurposing the drug. They are going to use against various targets. So here the company name you can see which company and which is the commercial name, which year it is launched and against which target. Okay, who introduced that one. So many you can see actually. So here you can see the GlaxoSmithKline. Okay, so here is Pfizer. Okay, so these all the drugs. They have launched pure computational structure based drug discovery. Finally they have see all these drugs are using in cancer, hypertension. Okay, and similarly you can see SDIA. Okay, so SIDA and lymphoma. Okay, so many, then glaucoma, influenza. Okay, so many diseases, and now you are having so many things. And if you see another things called if you, in addition to the journal, initially I showed lot of, within 2-month publication I have seen everything is computational biology for COVID-19. Okay, now another thing called print. There are a lot of preprint servers. You see the preprint servers. So many preprint servers. Before preprint servers means whenever you are communicating, before they are communicating, before they are submitting any stage of their stage, they can deposit that in the form of a in a database with an authenticated number, and that is permitted by most of the publishers as well. Okay, so all the preprint you will be able to see only drug discovery, computational modeling and drug discovery papers. So that is what I wanted to tell you. So this is what I have told. This very recently one particular paper we have published, critical reviews in microbiology. This is what overall things I have told. I have told actually. So here you are going to analyze very critical literature review. You are going to analyze the problem. Okay. So you are going to screen certain bacteria or certain virus, for example COVID-19. Okay, their novel gene you are going to identify. Where will you take? You will take from the relevant database, and you will identify the non-homologous human target. Okay, always you should identify non-homologous human target, because the drug, if you identify the same target is there in the human, it will not go and bind it to the target of the organ. It will go and bind it to the human target. So it should avoid non-human homologous target. Then you should identify, I told you, the metabolic pathway. What is the metabolic network of that particular gene? Which all are the important proteins? Identify that. You are having beautiful pathway database like a KEGG. Okay, so you can have the unique pathway you can identify. Then inside that particular pathway, which location that particular protein is identifying, that is present. Then finally you will identify lead molecule from a very big pool of data from literature survey or some natural ligand molecules, or say for example a lot of database like ChemSpider, PubChem, okay, Zinc database, Supernatural 2, okay, all those databases are there. So they have a lot of compound libraries, or you are having your own compound libraries, or literature survey some natural compounds library, or you may have a plant extractor. You might have extracted some compounds and MS data you might have. Okay, you can draw the structure and you can convert it into 3D structure. You can make many molecules, and what you need to do, as I said, you have to do the adsorption, distribution, metabolism, drug likeness. Okay, all those things you will do the literature survey. Then all those things you will do. Then this particular part, literature survey and everything is done, and ligand is ready, target is ready. Then again there are two aspects. The structure is available or not. This target is crystal structure is available, it is okay. Crystal structure is available, then you take the crystal structure. Okay, then take the crystal structure and do the docking studies. If crystal structures are not available, then you will go for the hypothetical modeling. Okay, either homology modeling you will do, or fault recognition or ab initio, depending upon your need. Once you model whatever I have told, you are retrieving the target, best template selection, structural alignment, superimposition, model building, evaluation. Finally this one. Okay, lot of things. Say for example a project you will check like Ramachandran plot. Okay, so many things you will do, and all analysis you will do, actually multiple analysis. Finally that particular model you will minimize, energy minimize, and predict the binding pocket of that particular model, and you will identify that particular model, and you will do a molecular docking studies with the best molecule, and you will be having the docked pose, best docked pose. So many molecule you have screened, only one or two will come here. Finally you will do the MD simulation, and one best pose you will get. That best pose you will go for a experimental studies. So this is the overflow of a computer-aided virtual screening. Now I would like to give few things what currently I am working. Actually, you know that I basically a microbiology person. I am working on antimicrobial resistance microbiology, especially priority pathogen like Acinetobacter, Pseudomonas. Okay, similarly Helicobacter pylori. Okay, Listeria monocytogenes, Staphylococcus aureus. Okay, now currently I have started many projects with so many work with COVID-19. We are having COVID-19 target. Okay, very recently published in Journal of Biomolecular Structure and Dynamics. Here carbon fluorine, okay, acts as the potential lead molecules against the prospective molecular targets of biofilm producing multi-drugs Acinetobacter baumannii. Acinetobacter baumannii in 2017 WHO declared as the highest priority pathogen. Carbapenem, the last line drug of an antibiotic is carbapenem. That meropenem, doripenem, imipenem, ertapenem, okay, pharopenem, and the colistin. If there are any bacteria they are resistant against these two antibiotics, then it will be very tough to cure actually. So that particular resistant organism, Acinetobacter baumannii, hospital settings, a lot of clinical settings that is there. So that organism WHO declared as the priority pathogen. This is a paper, only a pictorial representation of important images I have shown here. See, here I have identified the important metabolic pathway. Okay, so many genes you have identified. That particular gene we have created a metabolic pathway by using string and cytoscape. Okay, then what you have done is, then we selected the target. Some target, so many targets we have there. 4 targets we have prioritized. Then one or two targets we have selected, and one target, some target crystal structures were available. Okay, so for example, outer membrane protein. Okay, crystal structures were available. Some of the target crystal structures are not available. Then docked with the carbon fluorine, how it is binding. Okay, then after once it is done, the stability of the binding process we have tested by using molecular dynamic simulations. Okay, so you can see an excellent result, and this can be go for the further experimental evaluation. But still, that the toxicity and other problem related to carbon fluorine, the use as a drug, need to be further investigated. Then very recently another paper, it is published in Infection Genetics and in Evolution, published by Elsevier. So here we are proposed as steroid natural episterol from a fungus. We have identified from a fungus, and we have again same multi-drug resistant Acinetobacter baumannii. We have got some insight in silico modeling. Further we have done for in vitro data. Here again the first step is target identification. Major molecule we have identified, and the network individual proteins involvement with other proteins we have identified, network pathway we have created. Then what we have done is we have modeled some of the protein that doesn't have 3D structure. 3D structure is there, then we have taken that. 3D structures are not there, we have modeled. Then we have done the modeled protein, we have validated, proved that the good quality structures. Then we have docked against with so many target and one particular potential molecule, natural episterol, that is showing a very potential activity, best binding process with all the thing. And the MD simulation result I have not shown. You can refer the same paper, and based on that result we have done some in vitro investigation also, that also has given promising result in concurrent with our studies actually. Then again very recently a fungal yeast, Candida albicans, you know that mitogen activated protein kinase COG1 gene and cell division control protein CD42 are the putative targets of for natural molecule. There again you can see we have created the network. You can see the 3 important target that is a very play a central role. You can see that this has interacted with many protein. Obviously if I block with this particular protein, I can come up with a metabolic, I can come up with a drug screening insight. Okay, so I have selected that. Then crystal structure is not available, so obviously modeled and validated that. Okay, we have more and docked. Okay, and you see that high stability, docked complex, and energy minimized. Then similarly MD simulation, so done experimental analysis, it can be done actually. Similarly, when Zika virus infection, this was published in 2017, some of my students. So Zika virus surface protein, again like corona, the virus has the non-structural protein. Many of them doesn't have the structure at all. So structure, better the molecular structure, we did not have structure. That structure we have predicted. Then hypothetical model we have made, and we have selected some molecules and done modeling molecular docking studies. Then we have done the dynamic stimulation studies and identified the best pose, and done for the this one. Since all the things are viruses, animal studies are required, you know that, but still it will give a lot of things, and all these papers are gone to there's a high impact factor journal, Frontiers in Microbiology. Then similarly, when the time of Ebola, myself and my students were worked Ebola protein. So we have identified some of the proteins, and you know that these are the proteins in Ebola, target proteins, and that is again very active role that will perform in the Ebola virus. And that some of the protein has did not have structures, some of the protein have structure, and whatever it is shown here, it is a binding site we have predicted, and some lead molecule using some database we have identified, and we have shown that particular database over here. Okay, and best docked process here, and the MD simulation we have done, and that we have reported in 2016 computational life science. Then again multi-drug resistant Salmonella typhi, the same fluid we have tried the activity, how it is functioning, and we have seen that the network, whatever the important protein that is involving the fluorine, has an excellent binding properties, and it has done some molecular docking studies, and we have done
some ideal dynamic simulation studies as well, and that is reported in this particular paper actually. So another one more thing, since I was very fond of fascinated Dr. Bhavamani, again my study, I have so many papers on fascinated Dr. Bhavamani. There are the entire the drug resistant mechanism, how the bacteria become drug resistant, that particular pathway we have retrieved from KEGG database, and a major important target. You can see 5 different targets we have selected depending upon their priorities and their role in these particular things. We have selected, or if we block, say for example, ribose phosphate pyrokinase, shikimate dehydrogenase, aspartokinase, aspartate aminotransferase A, all those proteins are critical virulent factors, so they are taken as the target. No structures, so we have modeled, okay, we have identified natural molecules, and we have done some docking studies, the best docking studies. You can see the binding energy, they selected the result based on the binding energy and number of hydrogen bond, etc. And you see, and we have compared our result with one of the natural molecule to against all the putative target, how the binding energy minus 9.8, minus 9.8 is high binding energy. You can see excellent binding energy. These are the control in comparison with the how the approved WHO approved drug clinfloxacin, and that is the target is topoisomerase 2, and imipenem, last drug antibody penicillin binding protein. Okay, it has a binding energy of minus 5.9. Polymyxin lipid binding domain, it has minus 5. So you can see that our theoretical binding energy is much more better than that of the experimental binding energy. Thus what we can do is there is a high scope that this particular molecule can be used against this particular target, and further studies can be conducted. Similarly, this study we have in collaboration with one of my students, in collaboration with the NCBS Dr. Saudamini Madam's lab, prolyl oligopeptidases, so Shivanella woody, so that particular bacteria, it's a very specific type of bacteria, and we are she did a lot of work actually. So then you can see here protein modeled, and the best quality proteins identified, and the energy minimized, and comparatively come up with the good models and some docking studies, and some natural molecule like all that is reported, vinblastine, vincysteine, all these molecules are having excellent binding property that we have reported in 2015. And again I told about Listeria monocytogenes, that is a fungal that is a bacteria that has even is a foodborne pathogen, and there are 2 important proteins are the SEC proteins, that is the target proteins. That particular target protein doesn't have structures, and we have identified the folding patterns, and there is only a very less amount of difference. It is there, SEC K and SEC P, and we have done some docking studies with some molecules, and we have got a promising result, and again JBSD it is published in 2015. And again I have worked on multi-drug resistant organisms. Again this is a DHFR, the structure of DHFR, the multi-drug resistant gene produced by Salmonella typhi, that is dihydrofolate reductase, that gene responsible for the one of the conventional drug of typhoid endemic fever. That drug is known as the trimethoprim, okay, methotrexate, okay, trimethoprim, okay, trimethoprim. So this trimethoprim drug, we have predicted that we have identified the this was we have isolated the bacteria, sequenced the genome, and that particular genome what you have done is I have converted that nucleic acid structure into amino acid structure, and made a hypothetical model of that particular protein, and finally we got a good model. You see there is nothing, high quality model. Similarly, some molecule like resveratrol, now the very well studied molecule, and voginine, another 2 important molecule showing a potential binding with this particular target of DHFR. Similarly, another important organism is vancomycin resistant Staphylococcus aureus. Staphylococcus aureus, 1H protein. Okay, MRSA, all of you know that. But now after MRSA, the VRSA, the last line drug, vancomycin also become resistant. And I isolated a particular protein from some source, some bacteria, and sequenced that particular things, and we have got the nucleotide sequence. And once you got the nucleotide sequence, you can do so many things actually. We have modeled, and again predicted the model. None of the things are structures, okay, so we have modeled and energy minimized, and all the structures we have deposited actually. And we can see some molecule like luteolin, okay, so taraxacin, and all those things are showing some good binding interaction with this 1H of a VRS actually. And similarly, again as I said, in the streptolysin also the multi-resistant toxin produced by Streptococcus pyogenes, okay, Streptococcus pyogenes, and we have identified the streptolysin toxin, okay, and that particular toxin, and we predicted the secondary structures and predicted the model, and the model is a good quality model, and we have identified some of the molecule, okay, so some natural molecule, and they are showing best binding energy actually. So why I am saying all the things are a natural molecule, because most of your plant based background or botany background, okay, so that you will be able to identify explore the activity of natural products, okay. So then I have not shown any experimental data, since it's a computational biology only. If you read the paper, there the data is there, and again we are identified some ligand, some natural molecule from the symbiotic bacteria that is associated from marine sponges. Here I have another paper on that actually. That marine sponges, we have identified certain symbiotic bacteria, and that symbiotic bacteria produces certain compounds, novel compounds, and that particular compounds we have we have identified the compounds, and that particular compounds are having excellent binding potential with some of the target in Ebola virus. That time Ebola outbreak was there, so we have taken Ebola target, and you can see that result, and it is how it is binding, and the energy minimized the structures gives a promising result actually. So in conclusion, I would like to conclude this particular computer-aided drug discovery, the heart of bioinformatics currently actually, the black box of a plane, like that we can tell, okay, the black box of a computer, box of biological science, potential vaccine or lead candidate you can develop COVID-19. Definitely we can see many papers will be coming, okay, and we can study the functional genomics, proteomics, metabolomic aspects, the molecular interaction studies of biomolecular pathways, okay, and the genes and protein responsible for the drug resistance we can studies, okay, and expression of various genetic traits, okay, the relevant enzymes and their three-dimensional geometries we can study, the probable drug target, comparing the enzymes and their novel faults, okay, the pharmacokinetics, pharmacodynamics, the what to tell, okay. So the drug discovery now currently we can complete the drug discovery, all the initial part we can do by computational biology, make the molecule in a very best screened molecule with ideal drug likeness, bioavailability, and okay, all those things. Similarly, novel probable drug target we can do, and novel domain architecture, comparative genome analysis, evolutionary basis of the evolutionary conservation. Now people are been using the genome of COVID-19, okay, you can see that they are taken the genome of genome identified from China, then it them to Italy, okay, then UK. They are comparing, and they are finding out the similarity, what is the differences, if they are arises from a common ancestor, from where is the origin, all those things. Such a kind of high level study can be conducted actually. So this is the thing actually. So finally I would like to express my deep sense of thanks and gratitude to the people. They have invited me for the program, okay, for respected Honorable Vice Chancellor Professor S.V. Halshe, okay, similarly Professor Baswaraj Bankara, okay, the Registrar of Academic Davanegere University, the Registrar of Evaluation Professor H.S. Anita, okay, Professor V. Kumar, the Dean of Science, Professor Gayatri Devaraja, Director, okay, Internal Quality Assurance Cell, okay, and the Organizing Secretary Dr. Tippaswamy Risdappa. Special thanks to Manoj Kumar Artikala and Kalpana Manjareddy. In fact, I had an interaction. So many times I got an opportunity to interact with both of them. Okay. So thank you. I would like to thank you today. My the coordinator of that particular program, my session was Dr. Quadrisha. Okay, I thank him. Okay, and similarly, I thank Dr. Halish C, okay, Dr. Manasa DJ, and Mrs. Nivedita BT for all the supporting staffs they have done when I have done the webinar. Although it was a lot of technical issues were there, okay, from this one. That is the reason I have recorded this particular things once again. So that is what, and it's my pleasure to thank my principal in charge, okay, my principal in charge, and other faculty members, especially my mentor Dr. Sarala Gopalakrishnan, and my HOD and my great teacher of my institution, and my PhD guide and my mentor. She is the person, she gave me a lot of confidence and trained me for all those the research line. I sincerely thank Dr. Rajeshwari and so many students from all over India, so many research scholars, so many faculty members, so many people, and during this lockdown time you have put a lot of interest to listen this webinar, and I thank all of you, okay, once again. And very importantly, and most importantly, I would like to thank the very special person to Professor Dr. Govinda Pasar. Okay, in fact, he was my colleague and my good friend and a great academician, and with a lot of research experience, but and with a very good with a humanity, and a humble person with a down to earth his nature, and I thank him as well, and he invited me for this particular program, and it is a great honor for me to talk in this particular program. So I sincerely thank Dr. Govinda Pasar. So once again I thank you one and all. Thank you very much. Okay, all the best wishes. Be safe, stay home, okay, and stay safe, and all the best wishes for you. And if you are having any clarification or any doubt, you can write it to my mail, okay, and you can contact me, and I will be always support you, and I am ready and I am open for any type of collaborations, and I work on computational biology with a lot. I took this thing as a passion, actually, computational biology. So that is what I want to tell you. So thank you once again. Thank you one and all.